Okay, good morning, everyone. I have the pleasure to open this first session of our symposium in genetics of Albanian human populations. And first of all, I would like to thank our scientific secretary, Anila Oda, for coming. I would like, in the same time, to extend our warmest greetings from our Center of Research in Biotechnology and Genetics, and from the organizers, and in addition to welcome the other colleagues participating in this symposium. In addition, now I see that here arrived our colleague from Albanian Parliament, Mr. Eduard Sansi. No, I didn't remember the surname because he has been also my ex-student of biology. And really we're honored to have even some representative from our parliament. As a matter of fact, I feel really very excited when I open this kind of symposium, because it is frequently I find myself, first of all, to my ex-students of biology that has a good career, overcoming a lot of difficulties. So really, I'm proud to have a lot of them here as an integral part of our activities. Well, I would like also to welcome the other colleagues that will attend our symposium from abroad online. The colleague from Italy, Raffaella Cascella, from Rome, and other colleague, Abdullah Alia from Prishtinës University from Kosovo, and obviously the other colleagues from medical genetics that are already integral part of our activities and our research. As a matter of fact, this day is really a special day because in the same day, in the same hall, in the same symposium, for 2 years, we received good news for the establishment of our Center of Research in Biotechnology and Genetics that was approved by our government. And really, I would like to take this chance to thank our presidency of Academy of Sciences for the support, for the strong support for this research center, and especially to our president of Academy of Sciences for his persistent support and personal support to the government for the establishment of the center. As a matter of fact, in the field of research in genetics, and especially in population genetics, we are living a new era, because, you know, a lot of techniques and methods are being applied. And first of all, by applying very recently artificial intelligence. Obviously, we have to apply the same advanced method. And for this reason, we call time after time to come with us the colleagues from the best institutions abroad. In this case, we have 2 colleagues, one from the University of Bologna, Professor Alessio Bottini, and from London College University of London, UK, that will give their excellent experience in the field of research in genetics of population. So we have to benefit from their experience, and that is why time after time we call to benefit from their experience. Well, now I have the pleasure to give the speech to our scientific secretary, academician Anila Hoda, to greet the opening of our symposium. Please, the floor is yours. Thank you.
Thank you, Professor Ilia. So, dear professors, dear students, dear colleagues, dear participants, as always, when I greet on behalf of the Presidency of Academy of Sciences, is a pleasure and is honor because I have the chance to welcome you here in the premises of the Academy of Sciences of Albania. But in a way, I am greeting also myself in this case because the symposium is organized by the Center of Biotechnology Research and Genetics, and I am member of that center. So beyond of the joke, so we are today gathered here in this international symposium on opportunities, challenges, and impact of collaboration in genetic studies in Albanian human population. That, as I mentioned, is organized by the center, but also by the section of Natural and Technical Sciences of the Academy of Sciences of Albania. Today's symposium brings together researchers, academicians, clinicians, and specialists from Albania and abroad to discuss one of the most dynamic and transformative fields of modern science, which is genetics and genomic research. The participation of distinguished scholars from leading institutions and the diversity of topics presented throughout this program clearly demonstrate the growing importance of interdisciplinary and international collaboration in advancing scientific knowledge. Human genetic studies are not only essential for understanding our biological history and population structure, but they also have profound implication for medicine, public health, personalized therapies, and the preservation of scientific and cultural heritage. In recent years, collaboration between researchers, institutions, and the international network have significantly expanded the possibilities for scientific discovery. Such collaborations allow us to share expertise, technologies, methodologies, and data in ways that were unimaginable only a few decades ago. For Albania, these developments are particularly meaningful. Genetic research related to Albanian populations contributes not only to international scientific knowledge, but also to a deeper understanding of our own history, diversity, and health challenges. At the same time, these studies open new opportunities for young researchers, strengthen academic capacities, and encourage integration into the broader European and global scientific community. However, alongside these opportunities came important challenges. Ethical consideration, data protection, equitable collaboration, scientific integrity, and the responsible use of genetic information remain central issues that require continuous dialogue and careful reflection. Scientific progress must always go hand in hand with ethical responsibility and respect for human dignity. The Academy of Sciences of Albania remains committed to supporting high-quality scientific research, promoting interinstitutional collaboration, and creating platforms where ideas, experiences, and innovations are exchanged in an open and constructive manner. This is also one of the main roles of the Research Center for Biotechnology and Genetics. Although modest in size, I mean in the number of researchers that are part of this center, the center has undertaken several major projects that has already outlined the principal directions of its scientific activity. Time has shown that the establishment of this center was a necessity. Professor Mjekësisë mentioned that it is already 2 years that the center has started to function officially as a permanent center. While its further development and reorganization of the center in a broader One Health Center, bringing together several life science institutes, represents one of our main challenges for the near future. Scientific activities such as this symposium of today play an important role in strengthening academic dialogue and fostering new partnership, serving the broader objective of defining the most effective pathway for realizing this ambition in accordance with the contemporary scientific standards and societal needs. I would like to express our sincere gratitude to all organizers, speakers, moderators, participants, and collaborating institutions for their valuable contribution to this event. Special appreciations go for our international colleagues whose participations enriches these symposiums and strengthens academic bridges between our institutions and countries. It is a kind of science diplomacy. I wish all of you a productive symposium, fruitful discussions, and successful collaborations in the future. Thank you for your attention and welcome to our home, Academy of Sciences of Albania.
Okay, thank you, Academician Hoda. Now I have the pleasure to give the floor to Mr. Shalsi from Albanian Parliament to greet our symposium. Please, the floor is yours.
Well, good morning, everybody. Dear Professor, it was with a great pleasure that I received your invitation, but I was not sure I was going to make it till the end, but here I am. And it's a great pleasure to see the old professors of mine, Mr. Mariglen and Mr. Besnik. Well, there is a young generation here, and Teklela, right, Valbona. There are quite a few people who know that I was graduated in biology and chemistry, but I'm not either a biologist or chemist. My life took a different direction. But in the last years, I've been in touch through Besnik Bare, ex-member of the parliament, very good friend of ours, with what's going on. And congratulations for establishing the center. We know that it's a small step, but for us, a significant step. And as a matter of fact, the Academy in the last years has been not just promoting itself better, but managing itself better. And he is the General Secretary, yes? Vice President, right. I'm not updated with the titles yet. But I'm not going to say too many words, but the establishing of a center is a good and important sign. And as I said, a small step, but on the right direction. And we know that more funds are needed, but now that the Academy is better organizing itself and we're seeing results, I will do my efforts in the commissions or in the Parliament of Albania. The other thing, Professor, is that in last days, some news have been spread it over about an analysis, and we should be sure that people get it right. I've been on different tables, even in the Parliament of Albania, and we need to better understand, because better understand, better support you're going to have. And thank you very much for the professors from the Italy and UK. If there is any other one, I don't want to forget. Thank you for coming. Albania is, after many, many years in transition. I don't know whether it's still in transition or not, but it's becoming a very good attractions, not only for tourists, but also for investments. So this time came as professors, but you're more than welcome to come, even as tourists, not only as researchers. We went from 3 million tourists to 12 million tourists in quite a short period of time. So we're making our efforts, and there is a good momentum, I would say, for Albania. And if we do our job correctly, we're going to have Albania also in the EU Union. Dear Professor, thank you, and I wish you a productive symposium, which I'm sure is going to be. And I see some young students. These professors were the heroes of our times. So profit as much as you can from them. Thank you very much, and wish you successful. Thank you.
Thank you, Mr. Salsi, and for the warm words. And we hope to have, as always, the support of the Parliament in order to, for the future, to have a higher budget, because, research needs some budget, and we started with a really very small one. However, we had some increased steps. Okay, now I would like to start with the presentations, but before, some very short information. We have two invited speakers, the first two professors, and they have 20 minutes time to present their presentation. The others have only strictly 15 minutes. So please respect the time we have for our presentations. I will start with the first presentation. Professor Gareth Hellenthal, as I told you, from University College of London, that will present leveraging genome data to infer human history. Professor, the floor is yours, please. Forward, backward, and this is the pointer.
Okay, great, thank you. Thank you, thank you very much for inviting me. So I've only been in Albania for a few hours, but I can see that from what I've seen around here, it seems like it could be a very good tourist destination. So it seems very lovely what I've seen so far. So I'm looking forward to seeing more. And thank you for the introduction. So I'll be talking about leveraging genomic data to infer human history. I've added a couple keywords, as you can see here, to the titles that you have in your program. So I want to point out that I'll be talking about autosomal DNA, so not the Y chromosome or the mitochondria, but everything else. And so the analyses that I'll talk about here are things that you could do with, say, whole genome sequencing data or SNP array data, so like hundreds of thousands of SNPs and genetic variants. And I'm going to focus on intermixing specifically. And so I'm just going to go through a list of things that my group and colleagues have been working on that you can do with autosomal DNA to learn about history, focusing on intermixing. Lots of other people are doing interesting work on various things as well, which I don't have time to talk about, but I'm happy to take questions on that later as well. Okay. Yeah, so as I said, I'm going to focus on intermixing events, which is also known as admixture. And so this is when two populations or more intermix at some time in the past. And what happens to your autosomal DNA is at the right here. So when the two groups mix, I'll represent one group with red DNA and the other group with yellow DNA. In the first generation since the mixture, if this child has one parent from the red population and one from the yellow, all along its autosomes, it'll look like this. It'll have one red chromosome and one yellow chromosome. And then this person's going to pass along a chromosome in generation 2 to its child. That's a mixture of the red and the yellow. And that's because of recombination occurring during that generation. And so what happens is as these recombinations pile up over time, our generations later, the population's autosomes might look like this. And you can ask, what's the probability of going from a red segment to a yellow segment down here, our generations later? And it turns out that if you make a simple assumption of random mating of groups, you get this relatively simple equation where the probability of going from a red segment to a yellow segment versus the distance between the segments, which is something that we can measure, so that's something that we know, is equal to alpha, where alpha is the proportion of DNA that comes from the red population, times 1 minus alpha, which is the amount that comes from the yellow, times 1 minus this exponential function that depends on R, which is how long ago the two groups intermixed. So here's an example of what this looks like. So if these two groups intermixed R equals 20 generations ago, and 50% of the DNA came from the red population, and 50% came from the yellow, you'll get this probability curve here. So this green line is the probability of going from a red segment to a yellow segment versus the distance between the two segments. And so the idea is if we can identify these red and yellow segments, we can construct this probability curve and use that to infer alpha and 1 minus alpha, so the amount of DNA from each of these two groups, and also R when the two groups intermixed. And so we do this. So we infer these yellow and red segments using a technique known as chromosome painting. So we take the autosomal DNA of some group, say H4, and we compare its genetic patterns, these black and white circles, to groups all over the world. Okay, so for example, if I compare this one to these three different groups, I see that it has some matching patterns with the blue population and some matching patterns with the red population. And so we call this chromosome painting. We've got software that does this called ChromoPainter. And here we see that H4 seems to be a mixture of the red and the blue groups coming together. And if that's the case, we can figure out the probability of going from a blue segment to a red segment all over the genome and use that to figure out exactly when these two groups intermixed. So here's a real example. This is an example from real data from a Latin American. And so here we took this DNA, we compared it to groups all over the world, and we saw that some segments matched to Native Americans and some segments matched to Spanish. And if you look at the probability of going from a Native American segment to a Spanish segment in this person's genome, it follows these black dots here. So that's the data. And it increases exponentially versus distance, just like that theory I just showed you predicts. And so we can fit an exponential distribution to it in red. And the rate of this curve suggests that these two groups, the natives and Spanish, mixed 11 generations ago. Okay, and of course, we know historically that's true. So that's about 300 years ago. And that's when you had Spanish people coming in large numbers to the New World and intermixing with the native population. And so you could see this signature just in a person's DNA. Here's an example of another Latin American who not only has Spanish and native ancestry, but this person also had some Chinese ancestry. And so you can look at what's the probability of going from a Chinese segment to a native segment in the same person. And that follows this purple curve here, where the rate is not nearly as steep. And we predict that these two groups intermixed 4 generations ago. Okay, so Chinese and native groups intermixed 4 generations ago. That's also historically true. So in the 19th century, or starting about the 19th century, you had Chinese people coming to the Americas as well, largely working as laborers. Okay, so you can see these signatures in people's DNA. And so what we've been doing is we've been applying these to populations around the world and seeing where do we see evidence of these mixture events. And so, for example, we've analyzed each of these different populations here in the map. And in pretty much every one of them, we see evidence of these intermixing events. Okay, so we actually have a web page where we give our results for most of these groups. All of the intermixing is occurring within the last 4,500 years. And I'll give you a few examples of some of the things that we see. Okay, so starting with Latin Americans again. So I mentioned that in this Latin American study, we saw that some people had native ancestry, some had Spanish, there's also Chinese ancestry. These are all things that we know about historically, but there was one kind of surprise, which is that a lot of individuals throughout Latin America also have this DNA that matched to yellow and orange here, which is Middle Eastern in origin. And in particular, there's a lot of orange, which is matching to Sephardic Jews. Now, Sephardic Jews were a Jewish group in Spain. When Columbus was coming over in 1492 to the New World, at the same time, the Spanish Inquisition was going on. And so Catholics and Christians were persecuting all non-Christians, including the Sephardic Jews. It was illegal for the Jews to come to the New World, but it had been believed for a long time that maybe some snuck on the boats to come over to avoid the persecution. But because it was illegal, there's no records of this. Okay, there's no written records, but genetics was able to tell us that it looks like a lot of people came over because we see widespread Jewish ancestry across Latin America. And so this is a case of where genetics can unveil these sort of hidden migrations that we've got no other records of. Another example, so I did a few years ago some work looking at the genetic intermixing history in Britain. And first what we did is we clustered our individuals in Britain based on genetics. So in this map here, each dot is a person in our study, and the colors refer to genetic clusters of genetically similar individuals. And we saw a very fine scale clustering. So for example, we could tell whether a person came from this county or this county just based on looking at their DNA. And we were wondering, why is there such fine scale clustering? And we hypothesized that maybe one reason for this clustering might have to do with different migrations from Europe into different parts of the UK over the centuries. And so we tested this hypothesis by comparing the DNA of these individuals to these groups in Europe to see if they matched to different regions. And indeed, we found evidence of two major migrations that affected the UK. So these clusters here that have bits of red, the red is matching to people who live in Denmark and northern Germany. And those are the areas where the Anglo-Saxons came from about 1,500 years ago. And when they came, they settled in quite large numbers into England, Southeast England, which is where we see the highest amount of this red DNA. And we date it to just after these migrations. Now, the Anglo-Saxons didn't really go into Scotland, but if you look north into Scotland, you see that the red goes away and gets replaced by green. And green is matching to Norway, which we date to the times of the Norse Viking migrations about 1,200 years ago. And the Norse Vikings, they came this direction. They owned this island, Orkney Island, for about 500 years. And then they continued down the west coast where we see the most of this green DNA. Okay, so you can kind of track these different Viking migrations across the UK just by using modern people's DNA. And you can also see how it's led to clusters that we can see today. So it's affected the genetic structure of the UK even today. Another example, this is a final example in this section. So I haven't had a chance to analyze Albanians. I'd like to do that sometime. I did analyze your neighbors though. So this is a screenshot from this web page that I showed you earlier. And in Greece, we see a kind of interesting signal that I don't know if anybody'd have any idea what this is, but we see mixing related to Poland. So a Polish-like group dated to about 1100 AD. We don't really know what this refers to. So in this paper, we speculated it might be to do with the expansion of Slavic-speaking peoples, because we see a similar intermixing signal throughout Eastern Europe, dated to the same time period, but we don't really know what this might refer to. If anybody has any ideas, please let me know. And so that's all kind of, so that's all kind of old work. So I thought I'd spend the rest of my time talking about some recent things that we've been doing. Starting with, we've developed some new techniques to identify whether certain populations share the same mixture signals. So they shared across populations. And so, for example, in this paper, we analyzed each of these 4 populations in blue here separately. So Lezgin, Uzbekistani, Uyghur, and Hazara. And in each one of them, we found some amount of DNA, so it's about 45% in Hazara, 50% in Uyghur, 10% in Lezgin, that matched to present-day Mongolians. And in each case, so we dated that DNA contribution to a time period that really overlapped the Mongol Empire. Okay, so we thought that this probably is just a single instance of migration of the Mongol armies into these particular regions. But at the time, we had no way of formally testing whether these four populations share the exact same source. So we've developed a new technique that can test this formally. It's unpublished. And it says that, yes, it is the same source. There's one pink source here that's shared with all four populations. And this pink source was allowed to be any of these groups in pink and blue here in Eastern Asia, but it picks only one group, this triangle, which is Mongolia. Okay, so it really is quite strong evidence that there's this one migration. It all looks very Mongolian. Another example, so a few years later, we were looking at populations north of the Baltic, so in Finland, Sweden, and Norway. And so we're analyzing genetic clusters in this region. So each one of these rows here refers to a different cluster. And in each one of these clusters, we found intermixing with an East Asian source at different proportions, so about 11% in some Finnish clusters, et cetera, and at different dates as well, given on the x-axis. And so, for example, in Finland, they had the highest amount of East Asian, so up to 11%, and a date of about 60 generations ago, so about 2,000 years ago or so. And then as you move to the west, as you move to Sweden and Norway, the proportion goes down and the date becomes more recent. Okay, so at the time, we speculated that maybe this is a single source from East Asia that came in and intermixed with people in or around Finland. And then this mixed group continued west and mixed with other Europeans in Norway and Sweden. Because if that happened, you'd get a drop in the amount of East Asian DNA and the date would become more recent, which is what we see here. So we asked, is this the same Asian source in all of these populations? And with our unpublished technique, we said, no, actually, it looks like there's two different sources here. So in Finland, primarily, there's this one source in pink that's definitely East Asians. In fact, it looks like these two different Siberian groups here. While there's also this orange source that you see in Sweden and mainly in Norway that instead looks Turkish. Okay, and Turkish has a little bit of East Asian DNA, which is why we were getting confused before. And so here's a case where we think that there's two different waves of migration from different places in this region. Okay, so another thing that we've been working on recently is asking, similarly, are there multiple different dates of intermixing in a region? So for example, during the Neolithic transition, you had farmers coming from West Asia who migrated about 8,000 years ago or so into Europe, and they intermixed with the hunter-gatherers living in Europe at the time. Okay, but there's a lot of debate over how many different waves of intermixing there was, and if it differs in different places. So we developed a technique to try to ask this question, and here we used ancient DNA. So these are samples from the early to late Neolithic, from Ireland, Scotland, and England. On this map here, each one of these columns is a single ancient DNA sample, and these symbols and lines are our inferred date if you just analyze that sample separately. Okay, so these are our inferred dates for each sample. These are the radiocarbon estimates in pink here for each of these different samples, the radiocarbon dates. And we built a model that asked, okay, how many dates do you need to explain all of this data? Is this consistent with how many different dates of intermixing? And here our model said it was consistent with just a single date in red here, suggesting that there was just one wave of migration occurring around 4900 BC to about 47 BC. Okay, so here we think in Britain, there was only a single wave of migration during the Neolithic that intermixed with the hunter-gatherers. Now, in contrast, if we look at Portugal and Spain, so if we look in Iberia at the same time, we see evidence of two different dates of intermixing. Okay, so some individuals have intermixing that occurs around 4100 BC, while others have an older date that occurs around 5200 BC. Okay, so here we think there was multiple waves of intermixing into Iberia. And so you can sort of pick up, the complexities of these different migration events. Okay, the final thing I wanted to talk about, which me and other people are doing, is trying to infer archaic segments within human populations. Okay, so in particular, we know that modern humans when we left initially Africa around 50,000 years ago, we met up with Neanderthals and intermixed with them. Okay, and as a result of this, anybody with entirely non-African ancestry, so such as myself, we have 2% of our genome as Neanderthal. It's actually not modern human. Okay, and furthermore, there was additional mixture events where some people mixed with another group called Denisovans. I won't talk about that, but there's been a lot of debate over how many kind of waves of intermixing were there and the complexities of this process of mixing with Neanderthals, how many different groups. And so one way you can address this is you can take modern samples and we can extract the Neanderthal bits in our DNA. And you could do that in different parts of the globe and then compare those bits and see, are they the same? Are they different in different areas? And so, for example, if you look at what this plot is showing is, this is the amount of Neanderthal DNA that you see in Chinese people in blue from East, so these are East Asians, versus the amount that you see in Northern Europeans in red. Okay, so you can see that East Asians, such as the Chinese, have more Neanderthal than you see in Europe, okay, on average within their genomes. We don't really know why this is. So this could just be due to demography in some way. Some people speculate that maybe there was a second wave of Neanderthal mixing that went only to East Asia, and so Europe didn't get it. Others have said that maybe this is selection. So East Asians have favored bits of Neanderthal DNA because they've helped them adapt. And so that's increased them in frequency. We don't really know the answer, but it'd be helpful to get more of these segments from different populations, such as Albanians, say, and then just compare these things to see if we can learn more about these migration events and all these dynamics of this Neanderthal intermixing. Okay, so yeah, I've kind of, just to conclude, so I showed you quite a lot of things that you could do, all with autosomal DNA. So the first I talked about is inferring these past intermixing events. And we have software that can do this, if anybody's interested. And I gave a couple examples of some things that we've looked at. So we've been able to see these hidden migrations into Latin America, this interesting signal of Polish-like DNA in Greece. We also have software that can cluster individuals based on genetics. And we applied this to Britain and saw that there's these geographically fine-scale genetic clusters in Britain that seem to be, at least in part, caused by these different Viking migrations over a millennia ago. We've also developed some unpublished software, so that's what this asterisk means, that can identify whether different populations have the same admixing source. And so, for example, in Mongolia, in Central Asia, we see a single source of Mongol-like admixture, while in these populations in Finland, Sweden, and Norway, in the North Baltic, we see multiple Asian sources. We also have unpublished software that can infer the number dates of intermixing. And we infer, for example, multiple waves of intermixing into Iberia during the Neolithic, but only a single wave into Britain. And finally, a technique that can infer archaic segments within human populations. And one of the things that we're actively looking at is, why is there this varying Neanderthal integration across populations? What's causing that? Is it different groups? And a good way to get at that is, again, to extract these segments from modern human individuals. Okay, so that's all I have. Just to say, yeah, so that's all for me, just to give a few thanks. So these methods were developed in conjunction with these people here, with all of the software available there. These are some of the people that helped lead some of those studies that I mentioned and my funding bodies. And I particularly like to thank these 4 individuals, which are these students that did a lot of this work, either during their PhD or during their postdoc. And then otherwise, yeah, thank you very much, and I'm happy to take questions. Oh, okay. So I don't take questions yet, but okay, sorry.
Okay, thank you very much for your very interesting presentations. Really, we are always fascinated by the application of these kind of methods in genetics of human populations that time after time give some new data describing in details the migrations of human populations. That is why we are trying in our work, because the history of Albanian human population is still a white page, so we are trying to write on this white page the same history as you try to do with these very interesting methods. Thank you very much. Now, I'm giving the floor for his presentation to Professor Alessio Boatini from Bologna University, Between Italy and Balkans, Reconstructing the Genetic History of Arboreal Populations. Please, the floor is yours.
Okay. Okay, well, first of all, I would like to thank the organizers for inviting me here. In particular, Professor Michelesi, thank you very much. It's a pleasure for me. As you can see, the title of my presentation is deals about Italy and the Balkans, and in particular, the Arboreal, which are a sort of link between these two geographical domains, Italian Peninsula and Balkan Peninsula. What I'm going to show you is, let's say, a sort of review of several studies that we did some years ago under the direction of Professor Davide Pettener. I'm happy to mention Prof. Pettener here because he was the head behind all these studies. And it's also a bit, I would say, nostalgic for me because some of these studies are among the very first studies that I did when I was a PhD student. So I'm particularly happy of remembering and showing you these results. The Arboreal. As I think everybody knows, the Arboreal are an Albanian-speaking ethnic minority located in southern Italy and Sicily. It is one of the biggest, most numerous ethnic minorities in Italy. Of course, they are related to Albania because they migrated from Albania 5, 4, 5, 6. There are several migrations centuries ago. They still speak, at least most of the communities, they still speak their Arbrisht dialect, which is an Albanian language related to the Tosk dialect of Albania. In Albania, there are two main dialects, groups of dialects, which are Tosk on the south and Geg on the north. The Arbresh languages are more related to Tosk languages. As I said before, there are several documented waves of migrations from the Balkans, from Southern Balkans to Italy, starting from the late Middle Ages. Here you can see just the most important, I think, of these migrations. What is interesting to note is the fact that these migrations are a sort of on the wall, compose a sort of tapestry, because as you can see, very often there are several intermediate steps, not only in Italy, but also in the Balkans, because we know that some of these Arbresh groups arrived in Italy after intermediate steps in Greece, for example, Morea, the so-called Morea. Okay, this is the, I would say, current situation, the result of all these processes. There are at least still existing 41 Arbresh municipalities in Italy. southern Italy. The most important group is in Calabria, and particularly in the province of Cosenza, but there are other communities also in other regions, for example, Basilicata, Molise, and Sicily. The very first studies about these populations were based on surnames. Of course, surnames have a cultural origin, but this is interesting, their propagation tend to follow gene-like rules. In particular, in many populations, surnames behave like neutral alleles of the Y chromosome. Okay, so let's say that at the time, surnames were a very important source for many studies. Why? Because they still are, an abundant and low-cost information source for genetic studies. In fact, there was this, sort of criticism that called these studies the poor man genetics. But, however, they were interesting, and they also allowed to reconstruct the past, because, of course, we have surname information also from the last centuries. So this is an example of those studies about the Arbrecht. This was titled Ethnicity and Biodemographic Structure in the Arbrecht of the province of Cosenza. So here we focus on the province of Cosenza. That is the most important group of Arbrecht communities in the 19th century. So this is the setting, 19 Arbrecht municipalities and 5 non-Arbrecht Italian groups scattered among these 19 communities. Okay, there is some geographic organization, the Arbrecht of the Pollino area, West Crati River, East Crati River. And as for the results, this is a classical MDS plot based on surname frequencies, which summarizes the most important features, the most important structure based on surnames. And what we see is that the MDS plot based on surnames faithfully reproduces the geographic distribution of these communities. So geography is one of the main determinants of the geographic structure, the genetic structure of human populations. However, if we add the 5 non-Arbrecht Italian communities, what we see in these 3 is that they belong to a completely different branch of the tree. So ethnicity, in this case, separates Arbrecht from non-Arbrecht. So this is the second important message of this study. Then there is this study. This is another study based on surnames. Here, I am one of the authors, and it was one of my very first studies, 2007. This focuses on the Pollino area, so again, province of Cosenza. And the structure of the paper, the idea of the paper of the study is an expansion of the previous one, in the sense that in this case we have 10 Arbrecht villages and 9 non-Arbrecht villages. So the number of Arbrecht and non-Arbrecht villages is more or less the same. And here we have 4 historical periods, that is 2 from the 19th century and 2 from the 20th century. Again, an MDS plot, this is the most important result of this study, which shows, number 1, a separation between Arbrecht and non-Arbrecht along the second dimension of this plot, so ethnicity, and geography along the first dimension of the plot, from northeast to southwest. Okay, so again, these 2 factors appear clearly on the results. Additionally, each community here is represented by 4 points, which are the 4 historical periods. And what is particularly interesting in this case is the fact that the most recent period, the 4th period, is near the center of the plot, okay, the red points. This means that these communities with time became more and more similar to each other. And this led to the so-called breakdown of isolates in the second half of the 20th century. Okay, as I said, surnames as pseudo-Y chromosomal markers. So the next natural step was investigating the actual Y chromosomal markers. And the first study that we did with Y chromosomal data is this one, titled Linking Italy and the Balkans, a Y chromosome perspective from the Arbresh of Calabria. I think it was one of the very first, or the very first study to publish original data from the Arbresh population. Here we sampled 40 individuals from the Cosenza area, Arbresh from the Cosenza area, coming from 13 different villages and bearing founder surnames. And if I remember well, all different surnames. We did a bunch of markers, SNPs and STRs. We compared the results with reference population from Italy and the Balkans. And this is the main result. For the first time, we observed that first plot on the left, the SNP's point of view, haplogroup's point of view. We can see along the first dimension, this is a correspondence analysis, along the first dimension, the shifting, let's say, from Balkans to Italy, Balkan population and Italian populations. The Arbresh are located very near to other Balkan populations and not Italian populations. The same can be said about the second plot, which is a tree, where the Arbresh, again, are located cluster with Balkan populations and not with Italian populations. So I think this was the first time that we could demonstrate the fact that the Arbresh conserved very well their original ancestry, their original Southern Balkan ancestry. Next study, again, Y chromosome, but increasing our scope, so with more samples, most notably including also, in this case, Sicilian Arbresh and not only Calabrian Arbresh. These are the samples. So as you can see, this time we included many samples, more or less 100 Arbresh from Calabria, more or less 50 from Sicily, Arbresh from Sicily, and several comparison populations, both from southern Italy and Sicily, and from the other side, Albania and Greece. A few results from this study. From the SNP's point of view, it is, as you can see, it is similar to the previous study. That is, there is a cline, a sort of cline, where we can observe the Italian population here, very similar to each other. These are the Balkan populations. As for the Calabrian Arbresh, they are located more or less where we would expect, that is very near to the other Balkan populations. These are the Sicilian Arbresh. Okay, so their position is a bit different. Okay, we expected, at the beginning, we expected that they were very similar, but it is not the case. So the Sicilian Arbresh may have a different history than the Calabrian Arbresh, despite the fact that they share the same language. Okay, this is true also for these three, where we can see that Arbresh populations cluster with other Balkan populations. But look, for example, at Piana degli Albanesi, which is a Sicilian Arbresh community, which clusters instead with Eubea and Corinthos, so Greece, and not Albania, for example. Next one. This is an admixture-like plot. We call that admixture-like because it is not a true admixture analysis, because, of course, this is Y chromosome, not autosomal DNA. But the idea is very similar. What he did was first performing a model-based clustering and then calculate posterior probabilities with a discriminant analysis. And so in this case, each bar is a population. Most interesting results here. From one side, we can see that Arbres from Calabria cluster with Toscs. Okay, so again, we can confirm this tight relatedness between these two populations. Instead, the Arbres of Sicily, Sicilian Arbres, cluster with Greece, not the actual Albania population, but with Greece, which is interesting. Particularly this Greece component, this component from Greece is evident in a village from Sicily called Contessa Entellina. So it seems shared cultural linguistic features, but different, or at least in part different histories, genetic histories. Last study, this is the last study. This was a widest study. The actual aim of the study was not the Arbres, but as the title says, study admixture layers in Sicily and southern Italy in the whole context of the Mediterranean. So we didn't focus specifically on Arbres here, but nevertheless, some Arbres samples were included. So what I'm going to do now is just to extract from the paper, from the study, some of the results that are particularly about the Arbres. So as you can see here, we used a lot of individuals from several populations, among them 25 Calabrian Arbres and 23 Sicilian Arbres. Other interesting, it is out of topic, but other interesting Italian linguistic minorities, Greek-speaking ethno-linguistic groups. And from the methodological point of view, this time we used autosomal markers. In particular, the genotype. We used the genotype for this genotype ancestry kit, in this case, for this analysis. Okay, this is a principal component analysis, a classic principal component analysis. It is the one that focuses on the Arbres and the novel samples that were used in the study. What we can see is a sort of line here that goes from Albania to Sicily and South Italy, and islands from Greece, Grecian islands. In the middle, there is continental Greece. This one was one of the, I think, most interesting results of the study. That is the fact that there is a differentiation between continental Greece and non-continental Greece. As for the Arbres, they are located here, Arbres of Calabria and Arbres of Sicily. That is where we expected them to be, near Albania and near Greece. So, in a certain sense, confirming the previous Y chromosome studies. This is a plot based, a cluster analysis based on the statistic F3 outgroup. And again, we can see that the Arbres of Calabria, this cyan or light blue color, cluster with population from the Southern Balkans. Okay, so it is the another confirm of what we are saying from what we were saying from a few years at the time, while the Arbres from Sicily have a different color, an orange color. This color is more frequent, this cluster is more frequent in southern Italy and the islands, Greek islands. Okay, so again, this difference between Sicilian Arbres and Calabrian Arbres. This is another analysis, in this case based on haplotypes, using one of the methods that Garrett mentioned before, that is ChromoPainter, and then a clustering technique called fine structure. Fine structure yields a sort of tree which can be cut, I would say, at different points. And in this case, we can see that at 5 clusters, if we cut the tree at 5 clusters, the Arbres of Calabria start to show some differentiation compared to the rest of southern Italy. Okay, it appears this green color, which is, at this point, I know you could guess, the cluster which is more present in Southern Balkans. As for the Sicilian Arbrecht, they differentiate from the rest of the Italian communities only with 14 clusters, and they take a color, a cluster, which is present in Eastern Sicily, sorry, Western Sicily, in some communities of Southern Italy, and again in Greek islands, mainly in Greek islands, also in continental Greece, but mainly in Greek islands. So again, we have this confirmation of different history for these two Albrecht groups. Summing it all up, from the surname point of view, we have seen that in Calabria, at least in Calabria, ethnicity is the main factor differentiating among Albrecht and Italian non-Albrecht, while geography explains population structure within the two groups. So we have these two main factors. With Y chromosome, we could see that the Albrecht not only preserved their ancestors, their cultural and linguistic features, but also they preserved their ancestral genetic diversity as a result of, as we have seen, different contributions from different Balkan source populations and different degrees of admixture with geographically contiguous Italian groups. I didn't talk much about it, but there are hints that Sicilian Albrecht have more admixture with Italians than Calabrian ones, for example. And finally, autosomal markers confirm the Y chromosome results. Albanian-speaking groups of southern Italy display a recent shared ancestry, which can be traced to their putative Balkan source population. However, while Calabrian Albrecht confirm a clear relationship with the Southern Balkans, and in particular Albania, and particularly Tosks, Sicilian Albrecht are supposed to have experienced higher admixture with Italians, first point, and probably there are historical reconstructions that say that intermediate migratory steps and subsequent repeopling events from Greece. So there are hints that indeed in Sicily there were at least some repeopling events that involved real, actual Greek populations. And this could help to explain those observed differences. Okay, I concluded. Thank you very much for your attention.
And now for everybody who doesn't know me, I'm Adela Parola, hematologist at the University Hospital of Mother Teresa. And it's a pleasure for me to present Professor Gjergj Bojaxhi, a researcher of Albania and a politician also, to speak about two surviving Illyrian herds that today make up the majority of Albanian population. Please, Professor, the stage is yours. As long as the presentation is ready, I would like to present, to thank very much the two professors for the presentations, and thank you also for the part of Illyrian and for the part of Albanian mythology. My surname is Italian, but I don't know why in Albanian. I have to check about.
In fact, I had some questions for both of the previous speakers, but maybe we can do it at the end. So while they're ready, I'm using YFUL as an international tree for to show how different male lines in Albanian populations have spread geographically at a certain timeline as well. And I've chosen one single haplogroup, which is for the presentation, which is R1b, PF 7563. Under this clade, there are 3 different branches. And I'll show how these different branches under the same haplogroup have clustered in specific geographical regions of West Balkans, and how they have spread, especially in the last, at the end of the Roman period, early Middle Ages till today. Most of the results are from the Albanian project Rënjët, which the high resolution test we upload in YFUL. But of course, there are results there that also are not part of our project, but that we communicate with the people that have tested. We use mostly YFUL because it's so much easier in order to understand the past, to look at the male chromosome. Most of these social movements at the time, they were male driven. And the Y gives us much more clarity in terms of geographical distribution and the timeline, when these lines were branched out and how they spread geographically. And of course, with the DNA, it's a toolkit that has different tools. We just use Y more just to have this kind of clarity. But of course, it's not the only tool, as it was presented before. Autosomal tests are also a very handy tool to use in combination with the other. So in this case, we have, actually I'll present 3 clusters, not 2. This cluster is from South Albania. So this cluster is from South Albania. As I said before, this is haplogroup R1b, clade PF7563, and then subclade is that yellow one there. It was formed 3,000 something years old. I cannot see it from here. And then the TMRCA, it's 3,000 years, I think, or 3,000. So anyway, all of these results, if we look at not just the flag, but the region, these are in Korça region, Berat region. This Greek result is from the Gramoz region. It's just right on the other side of the border between Greece and Albania. And they also, the person that has that result has an Albanian identity, even though the flag was put there because he's physically from Greece. So in this case, it shows that in the last 1,050 years, all this cluster spread around what is today southeast Albania and a bit of northwest Greece. It is only found in this region. There are no similar results like this in, let's say, in Romania, Bulgaria, Serbia, Croatia, etc. So the cluster it's very regional in Albania. This haplogroup PF7563 has the highest percentage in the world in Albania. Of course, so the origin is from North Black Sea area. It's an Indo-European line. But it seems to have had a large migration effect in West Balkans around Middle Bronze Age. And in the Albanian population, it is found from the north to the south. So this specific cluster, based on all the results that the data we have, it shows clearly that this cluster was based in what is today Southeast Albania, the region between Berat and Korça. And then from there, it's somehow survived from the fall of the Roman Empire and the barbarian migration in the Balkans. And it was re-expanded during the Middle Age period. So this is one example when we use the data to explain the Albanian history and population movement and the creation of modern Albanian population. This is one of the clusters that shows that This line, we considered from the previous population, which were the Illyrians there, and it was local to that region in the last 3,500 years ago, and it has remained there. So all its spread is based on that region. The next cluster, it's so this is from the area that is today. This is the area that is today from the Dibra Mat region. This area we call Gegria Vjeter, which is the sort of the lower part of Geg region. Again, this cluster is originated in the early Middle Ages, and all of its results are either in Albania, in West Kosovo, in Montenegro, and in West Macedonia. There is a Macedonian result there, I think, further down. The Montenegrin result is that one. There should be a Macedonian one further down, and that is also from Struga. So again, this is a cluster from the Illyrian period, which survived originally in this area. I'm not giving the name of the Illyrian tribes because it's very difficult to find the borders where these tribes actually were. So I can say for sure that it is from the region that is today Dibra Mat Martanesh region. That's where this line originated or was clustered. But that's all. I'm only living at that level and not going further down. There should be other people who should specify who could be potentially the Illyrian tribes that were exactly settled in that area. So again, this cluster, it's 1,000 with a TMRCA of 1,350 years ago. And it is found today very broadly in the spread. It was during Middle Ages. So it has spread to lowlands Albania, to Shkodra, to Leja, to southwest Montenegro, which is the Macedonian result, and the other side of the mountain range that Albania borders with Macedonia. It is not found in Bulgaria, Romania, Serbia, or Bosnia or Croatia, which I'll show you another example of another cluster that is spread differently. So the next one is these, as I repeat again, these are from the same haplogroup, PF7563, and this is a different clade. And this clade, based on the research we've done, is based in the region that is today Kosovo or South Serbia or the old Dardania in terms of the Illyrian context. As you can see, this cluster has Bulgarian results, Albanian results, Romanian results, and this doesn't have Serbian or Croatian results, but there are similar clusters. To simplify the presentation, I picked this one. Based on the distribution, this cluster in the early Middle Ages was based in what is today Kosovo or southern Serbia. And then from there, it spread in different directions. And this is how this group of the population from what is today Kosovo spread in the early Middle Ages after the collapse of the Roman Empire and the Slavic migration, how they spread in a different pattern from the other clusters that we have in Albania. So in this case, from Kosovo, they spread in different directions. Some branches went to what is today Bulgaria, some in Romania, some have remained or have gone up north in proper Serbia and Bosnia and Croatia. And of course, other branches came down to Albania. In this case, the ethnicity of these Albanian results, or with the Albanian flag, are both Albanian and Vlah. So you have the results here that are from Kosovo, Albanian, Albania, Albanian. You have with the Albanian flag that are Vlah from Korça region, that have moved south. And then, of course, you have the Romanian and Bulgarian results. So what we notice, and this is a pattern with different haplogroups, it's not just with this particular haplogroup that I picked for presentation, is that the Y chromosome that was locally based in Dardania, they have a spread which is much broader. Actually, some of them you can find all the way to Hungary. The Hungarians have done DNA tests. The royal family that they have from John Hunyadi, which was one of the famous generals, is actually from the same region, same spread, from the southern Serbia, or Kosovo today. And this happened in two waves usually, this spread. One was from the initial Slavic migration, and maybe the first Bulgarian Empire, which cooperated with the Vlach a lot. And then the second wave is after the Serbian kingdom occupied Kosovo in the 12th, 13th century. There was again a big movement of population from that area. And this can be shown through DNA tests, Y chromosome, but also Noel Malcolm has written a very good book about Kosovo, and he also has documentation there that shows that there was a widespread movement of population from Kosovo once it was taken over by the Serbian kingdom of Raška. So I just wanted to present these three clusters to show how we use Y DNA to understand population movement in the late, let's say, Illyrian or late Roman period, and then modern Middle Age and modern Albanian context. And of course, you can break this down more. For example, most of this Albanian group here, which is like a cluster, is based on a specific tribe in northwest Albania, which is called Shala. So we have this result, which is not part of Shala, but is in the Shkodra region, but is not tribal affiliation. And then all the rest are the same cluster that are part of the tribe. So the whole valley there, the whole tribe is made of one single male line, which was expanded initially in that valley. And then during Ottoman period, where there was a lot of migration from the Albanian mountains to the plains, people from this tribe, as many other tribes in the region, they moved both to lowland Albanian regions and also to Kosovo, which also had a sparse population and was very partially repopulated from these mounting tribes 300, 400, 500 years ago. So again, this method we can use at different level of this haplogroup to show the Y chromosome movement based on geographical distribution. I think that was it, more or less. Thank you.
Thank you very much, Professor, for your great presentation. And now it's a pleasure and an honor for me to present the academic, Professor Ilia Mikarezi from the Albanian Academy of Science in Albania. He's going to present now YSTR haplogroups diversity and paternal linear structure of Albanian human population. Please, Professor.
Thank you, Adela. Well, as a matter of fact, we are trying now to understand rather better the genetic history of Albanian populations. That is a contribution to the genetics of the Balkan area. So this is why we have started this project. The main objectives are indicated here, so I'm going very quickly through the main objectives just to respect the time available. So we should use generally the statistical methods just to analyze the frequency of different loci. And at the end, we shall try to analyze the relationships among different districts where the samples were grouped, and most probably the relationships among subdialects. We have already 170 individuals or samples analyzed that belong to unrelated males sampled in different regions of Albania, but respecting the golden rule that each individual that was selected had the four grandparents, the parents, and itself from the same region. We analyze the genetic markers, STR, generally, And later on, we analyzed the frequencies of different alleles across the districts and subdialects. Whereas for haplogroups, we used this predictor called DevGen to identify the different haplogroups. Well, these are the regions and subdialects regions where we sampled our subjects. I'm going very quickly to the data that are elaborated by using these statistical analysis, so analysis of frequencies and application of principal component analysis to the groups we have analyzed. So these are some very general data about our samples that indicate the quality of our sample. These are some other data that belong to the data for forensic analysis. So I'm not stopping. So they are different parameters that belong to forensic analysis. You can see some in red and in blue values that are higher and lower values for different loci, the parameters from different loci according to these most common alleles. This table indicates the relationships between the number of alleles and the gene diversity for each loci. And we have graphically the same relationship. So we see that mostly these haplotypes have the higher number of alleles and the higher gene diversity. Now, going to the frequencies of different haplogroups. So you can see these that are typical for the Albanian population. My colleague now was referred only for these haplogroup. We have divided them in some subgroups. So I will describe very quickly the typical characteristics for each group. So the first group is composed from these haplogroups. And the first and the most important one is considered a characteristic or typical marker for of the Albanian population from the Balkan Bronze Age and the expansion and access marker for ancient antiquity. The second one is, as a matter of fact, reflecting ancient Neolithic or later Eastern Mediterranean connections. You know that we have these connections with other side of Adriatic Sea. The next belongs to older Neolithic or Chalcolithic migration patterns from Near East that mix with local populations. The next one, most probably, is a legacy of early Mediterranean contacts with or Roman era movements from other side of Adriatic Sea. And the last one, known as Berber marker, as a matter of fact, likely represent ancient coastal movements or medieval interactions rather than a significant component ancestry. The second group is this one with some lower frequencies. So the first, sorry. The first haplogroup is associated with the spread of Neolithic agriculture from Anatolia to the Balkan region. The next one, as a matter of fact, we find in studies that analyze ancient DNA that most probably is a pre-migration lineage, suggesting that it has been present in the region for thousands of years. The next one, is present in Albania, likely as part of the antique Roman era. rather than initial Neolithic expansion. The third group is this one, rather bigger, with higher frequencies. And I describe very briefly them. So the first one, first is a presence in Albania. Its presence in Albania is generally attributed to the movement of Germanic tribes from north, as Goths and Vandals, through the Balkans. The next one, as a matter of fact, is rare in Balkans, including Albania, but it is a signature of Western European Sardinian ancestry that could be considered a significant native Albanian, not considered a native Albanian lineage. The next haplogroup, this one, reflects a mixture of Paleo-Balkan ancestry and more recent early Middle Ages interactions with South Slavic populations in the region. The next, as a matter of fact, is a small frequency, does not form a significant part of the core of Albanian paternal ancestry. The fourth group is rather bigger, but with lower frequencies. This one is, as a matter of fact, typical Albanian. And the first one, as a matter of fact, typical represent a low frequency, late antique or medieval contribution to the Albanian gene pool from Mediterranean or Ottoman era movement. The next one is a younger subclade that likely moved to the Mediterranean and Balkans via Anatolian farmer migrations or later Bronze Age. The next one is represents an ancient, likely Mediterranean Anatolian contribution to the Albanian population. The next one is representing an Eastern Armenian Anatolian migration from that region that reached the Balkan region at these Copper or Bronze Ages. The next one represents early spreading of J2 lineage in Albania, possibly arriving from maritime routes in antiquity. This one is found in Albanian and likely entered in the region alongside or following the Neolithic expansion, representing early culturalists or early Bronze Age population. This one is really typical for Albanian population as high frequency lineage found among Albanians that had its roots in Bronze Age Balkan region and is considered a marker of Paleo-Balkan population. The last one is this one of three members. The first haplogroup, as a matter of fact, is generally regarded as a later intrusion, likely associated with Slavic migrations. As a matter of fact, R1A, as was discussed by my colleague, is considered part of a long-term multi-way Bronze Age extension in the Western Balkans, rather than solely a result of early Middle Slavic migration. The last one is considered prevalent in Albanian lineage and identified as primary marker of the Yamnaya culture, similar with pastoralist movement from the street. This is a preliminary comparison between North and South of Geg and Tosk. So you can see the same markers, the same haplogroups with nearly the same frequencies, with the higher, some higher frequencies in southern region, in Tosk region, because of the relationships with the other side of Adriatic Sea. Here we have the application of principal components. As a matter of fact, we don't see any clear differentiation among North and South or geographical relationship. However, there is some trend that puts together these kind of relationships. More probably with the higher number of analyses, we can have most probably other results. This is principal component analysis to the relationship between subdialects. Even here, we don't find any clear differences between Tosk and Geg. However, there are some trends of this kind of relationship. And I'm going now to the conclusions. You can see here the conclusions about the analysis of the frequencies of Albanian, found in Albanian population. I can say in a few words that all these data we have presented witness the continuity of the, let's say, original, ancient parameters of Albanian population with the present actual Albanians of the population of Albania. So I have finished. Thank you for your attention. So,
So, for example, the ROA and the food is very clear, but very expensive. It's not around the growth line, how many I and one, and 253. Again, it's slowly for sure, it's not a timeline. But some of the other ones, the bigger examples, but some of the other ones are even R1D and 59. You found out a brownie of that location. So the majority from the old European anti-Europe. There are subplates that actually are from the place one period. So I think congratulations on the sample. but I think the resolution is to go somehow to very beautiful. Even for example, J2A, you see J2A branches that are from the Neolithic. And you see J2A branches that enter the use of the property. So, because classification is too broad, can be from different historical moments.
Yeah.
But we know because of the higher,
Yeah, you are right. So we tried here to present only the main haplogroups we were able to identify. However, we have even some other subclades identified, not presented here. And I tried only to describe very, very generally the main clades and subclades of the haplogroups. Yes, as a matter of fact, there are a lot of other subclades, and most probably most of them are not identified by our sample, because here we are only identified 151 haplotypes. So you know the number is low, that is not able to identify all the subclades, as a matter of fact.
But then as we got more higher resolution tests, we noticed the number of the L61, that these are really counted. And we don't keep those ABCs for a month or the down line. Same thing in the leaf line. If you would have all the cheaters, you know, and normally all the cheaters are leaf.
Yeah, yes.
One maybe one. But then it seems that under the two graphs displayed survived in Studimet. They were either denied that process, and then they entered development in the late one phase to organize in the people. So while they are in Europe, as well as China, also the big innovation. They are more late one phase than Europe. So you cannot get the information unless there is a
So another aim of us is not only to identify the fine, let's say, structure of haplogroups, but also to analyze the relationships, mostly the relationships among different groups according to 12 districts and subdialects. This is the first step. However, with the next steps, so with the if we realize in the future the whole genome sequencing, we can have a lot of results that could clear much better the Yes, but you know, the data from literature sometimes are not of the same timeline. So, yeah. No, no, I don't, I was speaking only in generally about their origin, not for the one of our sample. Yes, please. 5 minutes.
Repeat what I just. So it's hard to tell apart Belarusians and Polish. So in this particular example, yeah, when Polish was the most likely thing, but it wouldn't be surprising if it was Belarusian. We didn't have any Ukrainians as well. So in general, there was a general cluster of Polish, Belarus, and other Eastern European groups. They looked fairly similar. So I would, if I saw either of those, I would kind of give it the same interpretation. I don't think we can, geographically say it was Poland, for example. Yeah. Yeah.
Thank you. Mm-hmm. Yeah, probably to help, yes. Yes, yes. um, how can I say? The most prudent answer would be, most safe answer would be, I don't know. I hope so. I hope so. There is an idea. There is an idea of we have this idea of using some there is some unpublished Y chromosomal data, not only the samples that you mentioned before. There is some unpublished Y chromosomal data about Albanian and Irish populations. So the possibility of performing a Y chromosome-based study with higher quality, more than quality, with a higher number of markers, indeed exists. Indeed exists. They are still unpublished. I discovered a few weeks ago that this data exists. I didn't know. Yeah. Mm-hmm. Yeah, I agree. I agree. I agree. Private level. Mm-hmm. Okay, so let's cross our fingers.
Okay,
It, yeah, it doesn't, for what I know, it doesn't seem so. They seem to have a low level of Slavic admixture than Calabrian ones. Yeah, probably.
so we
Probably.
can continue the discussion now, even in now in coffee break, because we have a short coffee break of 15 minutes, so we can continue the discussion because the time is running so quickly, and the time is really very short for us. Okay, thank you, and enjoy the coffee break.
Thank you. No.
Did you, I had a question for you too. So yeah, you looked at everybody's focusing on white chromosomes, everybody looking at mitochondria. So for example, in the other, should we expect, so if they came from Albania to Italy, it was it only
Sorry, we lost some minutes for the interviews from the media. Now we are starting the second session. Now the floor belongs to Master of Sciences Armanda Deda, Increasing Discriminatory Power in Y Chromosome Analysis, Differentiating Y-filer identical haplotypes through Y-filer plus in a specific case. Please, Armanda, the floor is yours.
I am Armanda Deda, and I'm a student of doctorate studies. We started this study with the idea coming from the professor. He wanted to, for the first time, like officially, make a study about Albanian population. Since my field of expertise, it's DNA analysis and human identification. I'm a former forensic expert. I've been doing that work for 21 years. But the idea to study population based on YSTR and STR loci was coming from the professor. But today I'm going to present to you just a tiny part of the problems that we have faced during our studies. So we are going to study the population of 500 samples chosen in the population of Albania based on their inheritance. We wanted to choose people that are their parents and their grandfathers being born in the same place. So we wanted to have this kind of population and data. And we are going to examine or analyze the samples based on YSTR and STR loci. So far, we have gathered like approximately 400 samples. We are missing just 100 of them. And of males, we have only 174 samples. Anyway, in my forensic casework, the Y chromosomal STR, as maybe not many of you know, but my friends and colleagues might know quite well, that the Y chromosomal STR analysis is applied in two major categories of investigation: sexual assault cases and also paternity cases. But either way, the YSTR analysis is not an identification method. It's just sometimes it's ambiguous, and it's called complementary analysis. It's not identificational one. However, unlike autosomal STR analysis, YSTR profiling is considered a lineage-oriented approach rather than individual-specific method. But still, for our study, it becomes a quite tool for the purpose. Just a general information about the audience. So you know that we inherit from mother and father the DNA from them, but the YSTRs are transmitted and inherited from the father's side. This means that all the males in the family, like father, grandfather, and son, and uncles, and all males at that family, should have the same YSTR profiles. Whereas the mitochondrial profile that comes from the mitochondria, which is inherited from mother's side, shows that all the females of the family, like mother and grandmother, maternal side, and your sister and niece and aunties, they all have to have the same mitochondrial profile. in here, I've just showed some schematic view of how the X and Y chromosomes differ from each other. You can clearly see that the Y chromosome is much smaller than the X. So also the information that it contains might be smaller, but still, even though it is a smaller chromosome, the information that it owns or has in itself, it's for our purpose, it's more than enough, because there are a lot of loci that we can take into an analysis, and their discriminative power is good. So YSTR profile, I've shown in here the YSTR profiles. I don't know if the audience is familiar with what kind of view has an STR profile, but this one, in this, in this specific picture, you have just the YSTR profile. As you can see, per each loci in here, these are the loci that are taken into analysis, and down there are the values per each loci, which are represented with only one allele. And you might ask, why? Why one? Why just one? But because you inherit only from the father the Y chromosome, and you only inherit one copy of it, and that's why you have only one allele per loci. But sometimes, even though you must expect just one allele per loci, sometimes you get value of two alleles in a loci, even though it is from a YSTR loci. But these kind of let's say mutations, or these kind of artifacts that are shown there, they are real, and they happen because of the multi-copy that we take in analysis from the Y chromosome. Because the Y chromosome structure itself is like palindromic and also segmental. And that's why it has a lot of DNA particles that are inherited in many copies. And that is why, for example, in the profile, it is shown with two alleles and not just one. So this is the reason for you to have that view of the Y profile. In our case, we used the same kit that we have been using in our forensic casework, which is Y-Filer from Thermo Fisher. And Y-Filer Plus, we had this idea to extend our analysis with Y-Filer Plus, only because as it is shown in the picture, you can see that from the on the first profile you have like 16 loci into the analysis, whereas with the Y-Filer Plus you get to have 27. In a forensic casework, or in the forensic point of view, the more you have, the better it is, the more to the truth you're going. So this was the same idea I had also for our study in my doctorate, because even though we had a sample of 174 unrelated individuals, male individuals in this case, we still got same profile, YSTR profiles on different males that were unrelated in any way. So we had the same exact with the Y-Filer kit, but when we used and extended the analysis on the Y-Filer Plus, we got on the same, on these two different individuals with the same profile, we got with the Y-Filer Plus kit, we got 3 differences. Even though they were like Y-Filer differs from the Plus with 10 loci, the differences that we got were only 3. And these were specifically in the loci in here, like mentioned there, like DYS 518, in DYS 449, and also DYF 387S1. Because these are the multi-loci added on the Y-Filer Plus kit. That's why we got these variation between individuals. But what does it mean? Like, how can we interpret this? The most probable interpretation in this case would be that two individuals likely share a common paternal ancestor. So you cannot undo or not take into consideration that they are still related in some kind of way. Because other way, then what was the purpose of examining all evidences with Y-filer? So they are related in some way, but became genetically differentiated over generation through YSTR mutation. And they are not necessarily two completely unrelated paternal origins. So the most important point in this case was that the conventional Y-filer panel lacked enough resolution to detect those lineage diversion mutations, whereas the Y-filer plus with additional rapidly mutation loci was sensitive enough to identify them. So this is exactly why rapidly mutation YSTRs were introduced into forensic genetics, and to differentiate males belonging to the same paternal lineage. So genetic, due to my former work, I'm always thinking in two different aspects. So I'm thinking about genetic population, but I cannot forget the forensic part of that. So from a population genetic perspective, the actual observations may suggest that you might be dealing with historical founder effects, restricted paternal diversity, geographical isolation, or persistent of ancient Albanian paternal lineages. And from a forensic perspective, it demonstrates why identical Y-filer haplotypes do not always indicate identical recent paternal relationships. So to sum up with, I put like 5 main sentences here. And I'm going to read each one of them. So the Y chromosome short tandem repeat analysis is widely used in forensic genetics, and that's a fact. However, its discriminatory power can be limited, and that is why we, so not we, but also the committees that make or that work on the kits, they always expand panels and add more loci in YSTR, but also in autosomal loci. As we know, there are kits with 32 loci, but for the Y-filer, they expanded it to 27. A key concept in YSTR analysis is the presence of multi-copy loci, such as the ones that we also detected the differentiation between the two profiles. So unlike single copy loci, which produce single allele per individual, multi-copy exists in multiple copies. Due to, I also said before, I already explained this, palindromic and the segmental structure. So as a result, they produce 2 or more alleles within the same individuals. These loci are highly informative, but require careful interpretation, especially in forensic perspective or cases. In forensic interpretation, profiles that are with Y-filer but differ with the plus indicate that the individuals share a common paternal lineage, but are not genetically identical. This is particularly important in structure or closely related population, where haplotype is more frequent. And to conclude with, I had a last slide, I don't know. Maybe there isn't. Oh, okay, not important. Conclusion was that this is a valuable tool for us, the identification of the Y chromosome based on the YSTRs. And it has shown so far very important conclusions, and that are also admitted by other studies made across Europe and also in the Balkan countries. Thank you for the attention. It's going to be me, the person who's going to introduce the next professor and doctor who's going to present her study, actually, in this case. I'm going to give the floor to Dottoressa Raffaella Cascella, researcher. Oh, okay, but we thought that we are going to have questions by the end of the session, but if only by the end of this session, okay? Or we can discuss it private. So La Dottoressa Raffaella Cascella is going to introduce to us online presentation titled Teaching Genetics at the Multimodal Genotype-Phenotype Correlation in PH2 Associated Inherited Retinal Dystrophies. I don't know if she is online or attending. Okay, okay. We can move to the next presentation, which is going to be performed by Professor Abdullah Alia from University of Pristina, Department of Biology. Teaching Genetics at the University of Pristina, Actuality and Challenges. We welcome Professor Abdullah. The floor
Thank you very much for the presentation. I hope you can hear me.
Yes, we can hear you loud and clear.
And hopefully you are also be able to see my presentation for just 1 minute. It's possible. So can you accept the request for sharing?
Yes, we will right away.
Yeah.
And
Can you see the presentation now?
You're on, yes. Could I kindly ask you to maintain it to 15 minutes, please?
Thank you very much. I will try my best. Thank you very much. Hello, everyone. I'm very happy to be with you today, and I would like to thank Professor Ilia and other organizers for this opportunity. As you can see from the title, my presentation today is slightly outside of the main thematic of the seminar. Therefore, I sincerely hope that it will complement our broader aspects of the discussion addressing a fundamental issue, such as teaching genetics. In my presentation, I will focus on 3 main issues. Therefore, it is structured as follows. The first part, it's about importance, trends, and challenges related to the teaching genetics. Next, teaching genetics at the University of Pristina, specifically at the Department of Biology, with a focus on study programs, curriculum, and organization. Then finally, a contextual analysis of the situation abroad as regard to teaching genetics, and then bringing it in the context of the University of Pristina. So when it comes to the importance, it has to be emphasized that genetics and genomics have transitioned from academic concept to integral components of modern daily life. It influences healthcare decisions, societal perspectives, and of course, understanding of human biology in general. Despite this significance, the last years have highlighted some gaps in genetics education and the need for more effective teaching strategies that enable to shift toward more active and more contextualized learning methods. So due to the central importance of the genetics, it is one of the courses which are very important, and it is applied to every branch of biology. So it is most important component of the most of biology curricula. Not only, but the focus of my presentation today is on biology curricula, and with more specifics at the Department of Biology, University of Pristina. General genetics is often taken after 2 semesters of general biology studies, and usually after 2 semesters of general chemistry. Among the main challenges that are evidenced everywhere, it is that the students very often come with varied background knowledge, and this is something that influences the course and the quality of the course. And another important point is too much information to cover in one semester genetic course. So it is usually like that. Again, the diversity in the level of the knowledge is also important, and always there is a run to keep a balance between the foundational material in genetics and new developments in genetics. In this context, one of the issues which is widely discussed is how to structure the material in the genetics curriculum, sorry. It is Mendel first or molecules first? In addition to that, there are also suggestions, there are also initiatives, there are also examples of more radically altered syllabi, like syllabi for genetics of the 21st century. I will give you an example which is published by Redfield in 2012, where syllabus which is differently structured, and of course it has also similar examples also elsewhere. It starts with personal genomics, and then a great attention is paid on the genetic variation throughout the syllabus. And I can say that the genetic variation is one of the keywords, and somehow it is really in line also with the importance of the thematic of this seminar. Another important issue which is under the discussion everywhere, it is about the textbooks or the learning resources. They are considered quite conservative, and there is, let's say, an opportunity that online resources need to fill this gap, but still need to be good processed or adequately processed. When it comes to a kind of summary of the actualities and challenges in teaching genetics, there are some key observations and conclusions in this regard. And they point out the need to bridge a diverse student knowledge backgrounds with the constraints of a single semester course. Then a need to find a balance between the foundational Mendelian principles and modern molecular developments. So in this regard, again, it comes to, let's say, to support an idea of more radical changes in this regard. Then textbooks, of course, and modern tools and effective teaching also are emphasized in the discussions about this issue. So modern genetics education has shifted towards broader active learning approaches. But this requires teachers to be well-trained facilitators, and those teachers should utilize contemporary resources and present structured outcomes to effectively teach in the post-genomic era. So I'm presenting in front of you a list of these issues, and then summarizing as a key point that recognizing the recognition that teachers are a crucial factor in this regard. When it comes to the teaching genetics at the University of Pristina, Department of Biology, one of the 5 departments as the Faculty of Natural Science and Mathematics. Going back in the days, in the 80s, there was one single study program for biology, 4-year studies, 8 semesters, and finally they got the professor of biology title, and they were entitled for teachers at the primary and secondary schools. Then end of 90s, there were 2 study programs, biology, which was mainly for the education, for teachers, biology teachers, and the second one, biochemistry and physiology, or well known as applicative one. From 2000 to 2010, we had 3 bachelor biology programs and 3 master programs, whereas recently, again, we have 3 bachelor programs and 2 master programs in this regard. The main characteristics in this regard, when it comes to the teaching genetics, is that in the older programs, the genetics was taught in the 4th year of the studies. It lasted 2 semesters, so in semester 7 and semester 8. Here, I would like to emphasize how the teaching was organized, because there is still kind of inertia in this regard. So it was, there were weekly hours 2+3 or 2+2. Similarly, it was also the situation with teaching genetics in other programs in 90s and actual programs. An observed change in this regard is that the genetics is not anymore at the end of the studies, but it is in the 2nd year or 3rd or 4th semester. Whereas other genetics related subjects, which are characteristics also for the study program, follow genetics, so after 1 or 2 semesters, like human genetics as an optional course in 5th or 6th semester, or environmental mutagenesis, which is an optional course for the ecology and environmental protection study program. Just to mention, genetics courses, of course, are offered also in other study programs, which are organized in other academic units of the University of Pristina, so other faculties. So I just mentioned, and now it's in front of you, that usually a study program like biology, for example, which leads to mainly, let's say, for teachers of biology, has an elective as human genetics, because there is a lot of human genetics content in the secondary school. Therefore, we reflected on that, and human genetics is offered as an elective in this program. From my experience from 1995 in lectures and exercises, the research or practical from the genetics was mainly in the field of genetic toxicology and environmental mutagenesis. First, with Drosophila melanogaster, estimating genetic loads in the population of all the species, whereas with the increased human and laboratory capacities, introducing new research methodology and specialized courses, being involved in more projects and offering more opportunities also for the students, there is a long list of the methodologies which are used in this framework of research of our group here, geneticians, but also they are offered very often with participation of the students in various research projects and in the framework of a diploma thesis. I'm presenting also an example of the syllabus from genetics, and it belongs to the approach Mendel first and then molecules. But when it comes to the integration of the research activities, there is a clear indication also in the of this syllabus, and it is acknowledged by the experts in the process of their accreditation, so we got good marks. So we got good marks, and we were continuously trying to address the challenges that came together with the trends and developments in this regard. Just a kind of extract from the expert evaluation or report of the expert team, which emphasized that the set of policies and regulations are in place, staff recruitment process, teacher training, and suitable teaching strategies are in place and or are applied. However, what I would like to point out today is that beside the common challenges and beside the assessment by the different, let's say, experts in the process of accreditation, which point out some of the problems, some other problems somehow remain undetected. And on the other hand, they influence a lot on the organization and also on the quality of teaching genetics, and not only genetics. One of them, the organization of the teaching, it is mainly one teacher or professor per course and one assistant. So it is how to say, influencing also the quality, because individual professors are responsible for entire general courses. In addition to that, often managing multiple subjects simultaneously. There are some other situations of engagement, let's say, even in other academic units of the same university. Also, important situation or interesting situation is as regard of the provision of a subject, in this case of genetics, in different academic units, where in each of them certain professor is in charge for the entire course, where there is a lot of similarity in the content of that, and somehow it is not always delivered with the best available quality across the academic units of the University of Pristina. Another thing, which again remains somehow undetected but still has an influence in the quality, is the need for synchronization of the theoretical and practical part of the course. So it means, so I mentioned that a professor and an assistant are in charge or competent to cover the content of a subject. One is for the theoretical, whereas the second one is for the practical part. And in a weekly organized course, so delivering 2 lecturing hours or eventually 2 exercise or practical hours per week, it's not easy to synchronize with the content delivered in the theoretical part. Further, some of the phenomena in genetics need to be, let's say, supported by research-based, experimental-based teaching and so on, which is very often difficult to perform because there is no block teaching option available or intensive option of the format in teaching genetics. Despite some, let's say, improvements in involving students in projects, and also when it comes to a limited number of them involved also in the thesis from the field of genetics, it remains challenging that every student can be involved at least in some of genetic experiment, and this is not easy because of the format of teaching. What I would say as a conclusions or list of conclusions in this regard is that the University of Pristina is aligning its genetics curriculum with international standards, offering comparable content, syllabi, and semester hours, so which are again comparable to other universities abroad. So this is something confirmed in the process of the accreditation. On the other hand, while facing common pedagogical challenges, UP also demonstrated strong commitment, again confirmed by the process of accreditation, and evidenced by improvements in research-based learning and practical laboratory capacities and teaching resources. Again, despite this progress, the university faces specific institutional limitations regarding staff appointment and teacher organizations. And what I'm pointing out today in front of you and comparing or analyzing contextually with latest developments and discussions about teaching genetics is something that one can see it from inside, and unfortunately, very often it doesn't come, let's say, to a table for discussion and eventually to address it to improve the situation. I hope that this case I presented might be useful for addressing the challenges also at other universities if the situation is similar. Here I would like to thank you very much for your attention, and if you have any questions, I'm more than happy to answer them.
Thank you, Professor, for your thorough insights. We must continue to the next presentations. I have confirmation that we now have online connection with la dottoressa Raffaella Cascella.
Hi, good morning. How are you?
Hello. Welcome.
Thanks. Okay.
I'm leaving the floor to you.
Okay.
Okay.
Can I start? Okay.
Yes, you can start.
Okay, thanks. Just an information, I may have some connectivity problem, so I will turn off my camera, okay? Okay. Questo.
Sorry, can I please kindly ask you to keep it at 15 minutes, the presentation?
Okay, no problem.
Okay, thank you. Thanks a lot.
Okay.
We are only seeing you now. You can go on with your presentation if you like.
Okay, okay, perfect. Perfect, thank you.
Okay, you're welcome.
Okay. Oh, sorry. Oh. Sorry. Okay, it's okay. So, first of all, thank you for the opportunity to discuss my research work today. And I will present a recent study conducted in the Italian cohort of patients affected by inherited retinal dystrophy. And it will be very interesting to replicate this research in the Albanian population to further investigate population-specific genetic architecture and improve the diagnostic and the precision medicine approaches. Okay, as we know, inherited retinal dystrophy are an heterogeneous group of genetic disorder characterized by progressive degeneration of photoreceptor and the retinal pigment epithelium, often leading to visual impairment and blindness, as reported in this picture. These different group of disorder differ in the age of onset, severity, and rate of progression, and inheritance pattern among patients. The prevalence is variable, and it is possible to recognize specific morpho-functional changes as reported here. So, inherited retinal dystrophies are caused by genetic variant located on different genes, and today these pathogenic variants are associated with different form of ocular disorder. And in fact, today have been identified a lot of pathogenic variants located in 300 genes. So, as reported in this picture, inherited retinal dystrophy display different pattern of inheritance. In fact, it is possible to recognize an autosomal dominant pattern of inheritance, autosomal recessive, X-linked pattern, mitochondrial, and multifactorial features. In case of ocular disorder characterized by multifactorial features, it is important to remark the interaction between specific variants and different environmental factors. In order, it is important to remark that this heterogeneous group of disorder are characterized by incomplete penetrance and variant expressivity that further complicate clinical diagnosis and genotype-phenotype correlation. So, and an example I would like to present is the retinitis pigmentosa. As reported in this picture, it is clear that this form of retinal inherited retinal dystrophy is characterized by different pattern of inheritance. So we can identify different forms of this disorder that may have similar phenotype, but with different genetic features. And in these slides are reported example of major causative genes involved in retinitis pigmentosa and other inherited retinal dystrophy. As you can see in this picture, some genes may be associated with different form of this disorder, highlighting the remarkable genetics and clinical heterogeneity of this disorder. So, among of this gene, our attention was focused on PRPH2 genes, and in general, pathogenetic variants located in this gene are associated with late onset form of retinal dystrophy. As reported here, this gene may cause different form of retinal dystrophies, and as we know, a pathogenic variant in PRPH2 genes account for 23% of inherited retinal dystrophy with an autosomal dominant inheritance. So, in this picture, is shown how a subject carrying PRPH2 variants may experience the surrounding environmental. And it is important to remark the degeneration of macula, and in particular the degeneration of cones. So, considering the clinical and genetic heterogeneity of PRPH2 related disease, genetic testing represents a valuable tool for supporting the clinical and instrumental diagnosis. The aim of this study is to identify disease-causing variant and link this genetic variation to specific disease manifestation. So we decided to create a multimodal approach to enhance genotype-phenotype correlation by integrating genetic, clinical, and instrumental data of our cohort of patients. So, in this picture is reported our multimodal workflow, and it is clear that in this case the first approach is the clinical characterization of patients, and then it is important to manage our patients with a specific genetic counseling that in this case is called pre-test genetic counseling. Then our cohort of patients was addressed to the lab analysis, the identification of variant are addressed to a specific genotype-phenotype correlation. And then another important element of this workflow is the post-test genetic counseling in order to explain the results to patients. And then all the results should be shared with clinicians in order to realize a specific genotype-phenotype correlation. So, in this picture is reported our genetic workflow, and first of all, our patients, our cohort, it will be addressed to the sample collection with the buccal swab or blood sampling, the extraction of DNA and the analysis of the genome by an NGS approach, and in particular using a whole exome sequencing. And at the end, another important part of our genetic workflow is the analysis and interpretation of genomic data. So then the aim and the element of this study is to identify novel pathogenetic variants located in the PRPH2 genes in order to expand the genetic and clinical spectrum associated with this kind of retinal disorder, and then improve the genotype-phenotype correlation. So, as reported in this picture, we have the possibility to identify then a pathogenetic variant located in our causative genes, and the majority of these variants are located in the exon 2 and exon 1. As reported in this picture with the different family tree, it is clear that the pattern of inheritance of the pathogenetic variants located in PRPH2 genes follow an autosomal dominant inheritance pattern. In fact, as reported in this different family tree, there are different subjects affected of the same condition with the same genetic variants. Another important results that we obtained in this study is the possibility to identify 3 novel variants not described in the scientific literature, and the possibility to confirm the presence of other genetic variants that are present in our genetic studies. As reported in this table, we have the possibility to correlate the genomic data with the clinical and instrumental information that we have the possibility to collect from the patients and from the clinicians. It is clear that this study emphasized the phenotype heterogeneity of this disorder, and highlighting how intra and inter-individual variability complicate a precise genotype-phenotype correlation. In fact, as reported in this table, we have the possibility to identify different variants that are correlate with different form of inherited retinal dystrophy with different clinical manifestation, even if the genetic variance is the same. So, a key strength of this study lies in its multimodal and integrated approach, combining genetics and ophthalmological assessment. I think that this approach is a good approach to finalize the results. So it is possible to create an important correlation between genomic data and clinical information. I want to thank you for the attention, and if you have any question,
Thank you very much for your presentation. It was lovely. We've decided that by the end of each session, we only have 5 minutes for the questions.
Okay, perfect. Thank you.
Thank you. So now, last but not least for this session, it's my honor to present Professor Eliana Ibrahimi from the University of Tirana with her presentation called Multi-Omics Data Integration for Cardiovascular Disease Diagnostic using the Bioconductor Ecosystem.
So shortly, oh, we are ready, good. So I'm associate professor at Department of Biology with expertise in biostatistics and computational biology. And my recent research focus is on metagenomics, data science, and integration of metagenomic data with other omics. Mostly applied for health problems. So today I will present work done in collaboration with a group at Leuven University, unit of cardiovascular and hypertension epidemiology, which aimed to classify cardiovascular disease patients based on multi-omics data. And in this presentation, I focus on the integration of microbiome metagenomics data with metabolomics data. So because I work on computational methods, the work is not actually experimental, but it's kind of laptop experiment. So we try to compare different statistical and artificial intelligence methods to see which one is doing better in classifying patients that have the different specific situation related to cardiovascular disease with a healthy group. So it's a benchmarking study, let's say, which in this case aims to compare methods that apply multi-omics, integrate multi-omics. Very shortly, I will keep my presentation in general short, but this is very shortly. So why microbiome and metabolome are important and how they affect the progress or maybe also the onset of this disease. So everyone knows microbiome is an important part of our digestive system, and not only, but here we focus on gut microbiome, and it is strictly related to the metabolites that we produce during food decomposition. But not only our metabolites, but also microbiome produces their metabolites. All these chemical, compounds affect the host transcriptome. This is a long topic. I will not really go in details. I'm not really expert actually to go in details on this, but the idea is that using this microbiome and metabolome data, we can see how then the host transcriptome or genome, also in some cases, is affected. So we do this in different ecosystems. We used the bioconductor ecosystem, which is one of my favorite ecosystems, and for students that are here, a great source to find packages that are doing a great job in analyzing genomics data in general. Bioconductor, just because I'm part of this community, so I will very shortly introduce, but not the aim actually of the presentation, is not a package, is an ecosystem which has thousands of packages maybe now, specifically addressing problems in the biological data analysis. So it's based on the R software, but a lot of things are also in Python now applied. These are some of the most important packages that are part of the bioconductor, which we use for the analysis. But it's not just the packages. There is a lot of other resources, training, workshops, conferences, and materials where you can learn all this stuff that is developed by the bioconductor community. So we are using this ecosystem mostly in the analysis for this work. And as I mentioned, we do computational work here, so we use open data from
studies that have made that the data public for researchers working on the data science field to work with them. In this specific study, we have used data from the meta-carotid cohort, a big European study that focused on the cardiometabolic disease spectrum. So we have a control group, we have a bigger group, let's say with ischemic heart disease, which has some subgroups based on specific problem. And we have data for the clinical lifestyle, other medication situation, the gut metagenome, shotgun sequencing data, and metabolome coming from serum and urine. So the aim is to integrate all this data to then classify the controls or the healthy people from the diseases. There are many steps and a lot of work and time to do all these, but very shortly, we first do feature selection. So these data comes with thousands of features. And then first you need to reduce a bit the features that you have, or species and metabolites, if I can say in practical language. And we do this, we use the lasso regression to do this, and then we have a sparse matrix with the selected features. Then we use the multi-omics factor analysis. It's called the MOFA2, a package that works in R and Python. So you have to integrate these two softwares to integrate this data together. And then at the end, you have a latent factors matrix, which you use to go ahead with your analysis or classification model. Very shortly, I will not do a mathematics lecture, but this approach, this method is just a generalization of the classical principal component analysis, but for multi-omic data. So the idea is that you have a big matrix and you want to reduce it and to have a smaller matrix, but which is now not with the samples and features, but you extract some factors which explain most of the variability in both the layers. In this case, we have 2 layers, but you can have more than 2 omics layers or type of data. And then we expect these features based on the layer to see which, and weights to see which of the species or metabolites are contributing mostly to the factor that explains most of the variance. Very shortly. After that, we get these latent factors. We decide we can have, for example, 10. So imagine from thousands, we go and pick up to 10, 20. It depends on the variability that they explain. And we perform, in this case, we performed applied 5 classification, machine learning classification models to classify deceased from healthy patients. And for all these, we then compare the accuracy and performance based on some matrix. Some results which I can consider preliminary. It's not nothing, it's definitive, but for example here, this is the plot that we get for the variance that is explained by each of the factors, which again combine the features that we have in the first matrix. And then we can see, for example, factor 1, what are the, in this case, metabolites that contribute to this factor. And we have seen in this case, for example, that the carnitine system is very important. Then this sign that you see here shows if the effect is positive or negative. And of course, the weight is showing which of these features is the most important one. In the microbiome case, we can see here different species that are important, like the some you see are contributing negatively, some positively, which means that some of them are absent in patients which have the disease, and some of them are more abundant in these patients. And this, we can see the effect or the difference between the factors also using these violin plots that we have. Compare the healthy with the diseased group. Then we use some factors. In this case, we select 10, but we can do more if we want to fit these classification models and then compare and see which is the best based on the accuracy, which indicates its area under the curve, to classify the disease with a healthy patient. And in our work so far, we have random forest as one of the best ones, but also this artificial neural network model seems to perform quite well. But still, we have a lot of work to do in optimizing these models, and say that, yeah, this is the final work we do. So we have, this is a work in progress, so we are planning now to apply other integration methods which could work better to integrate this data. And of course, which is what is very important, we need to then see how we can interpret these biologically pathways that contribute to all this, which is quite a more complicated thing to do. Yeah, this is all. I am very thankful my research stay and all this work is supported by Cost Action through Tiranës Cost Action. And of course, I'm very thankful to the group of Professor Tatiana Kuznetsova, which hosted me, and we are still collaborating. So that's all. Thank you so much. I hope it was short enough, and I think it's the end of the session. So if there is any question for me or previous. It's the end. Oh no. Oh, we have another one. Oh, okay. Sorry.
Now I would like to invite Gjenetikës that will speak about the study of MFFR gene mutation in the Albanian population.
Hello, I am Gjenetikës, and my scientific work is the title study of the MTHFR gene in a group of the Albanian population. In accordance with this purpose, I formulated also with objectives to identify the frequency of these two genetic polymorphisms, C677T and A129T, in a group of the Albanian population, to determine the genotypic combination between these two genetic variants, and to compare them with other populations to assess the genetic differences or similarity. To understand better the important role of this polymorphism, it's necessary to stop initially at biological role of the MTHFR gene. The gene that causes SI plays an essential role in the metabolism of folate and the cycle of homocysteine. It catalyzes the conversion of 5,10-methylene tetrahydrofolate into 5-methylene tetrahydrofolate, the active form of folate circulated in the blood. This form is essential for the remethylation of homocysteine into methionine, requiring by vitamin B12. The gene encoding the enzyme methylene tetrahydrofolate reductase is located on the short arm at position 1p36.3 and consists of 12 exons. The first exon, including a non-coding segment, called a 5' UTR non-coding, whereas the coding part of the second exon and continue up of 12 exons. MTHFR gene mutation, showing the C677T polymorphism, is located in exon 4, causes substitution of alanine with valine, in the catalytic domain, while A1-298 polymorphism is located in the exon 7, because this is substitution of glutamine with allin in the C-terminal regulator domain. In addition to their association with the cardiovascular disease for the C677T and A1-298 polymorphism are also linked to neurological and psychiatric disorders, such as depression and schizophrenia, metabolic disorder including type 2 diabetes and second type of cancer, autoimmune disorder such as psoriasis may influence health, increasing the risk of neurological tooth defect in this fetus. According to this objective set for the study, a detailed work plan, which implemented during the time period of 2023 to 2025, and included four main phases. First, collaboration with the medical clinic and collection of blood samples, selection of 149 symptomatic individuals for testing the C677T and A1-298 polymorphism in the anti-EGFR gene, molecular analyzing using the 7-3 technique, and statistical processing of the data with the PSS and Excel to assess the distribution of these variants in the Albanian population. For this study, the 7-3 method for the Vienna lab diagnosis was used to allow the identification of 30 mutations associated with the cardiovascular disease. Those samples were processed in 3 steps: DNA isolation using ITAGMT, amplification of ITCR, and hybridization with the 7-3 Result and discussion. As shown in the table and the bar chart for the C677T and A1-298 polymorphism, as shown in the bar chart for the C677T, the heterozygous CT genotype is the most highest frequency compared with the homozygous CC and TT genotypes. This indicated the modern effect on the activity of this enzyme functional. The finding that the mutant form T allele is mainly present in the population, but they are in the heterozygous form. As shown in the bar chart for the A1-298 polymorphism, the homozygous genotype for the A allele is most highest frequency compared with the heterozygous and homozygous genotype for the C allele in a group of the Albanian population. Also, the C and T allele was analyzing for the C677T polymorphism, while the A and C allele was analyzing for the A1-298 polymorphism. As shown in the bar chart for the C677T, the most frequent allele it was C, whereas the A1-298C, the most frequent allele it was A. This finding that the mutant form are present in the population, but they are not dominated. To evaluate whether the distribution of genotype for the C677T and A1-298C are in equilibrium, the Hardy-Weinberg was used implied. First, the genotype and allelic frequency were calculated followed by the expected genotype frequency according to the Hardy-Weinberg equilibrium. Using the chi-square test, the observed genotype frequency were compared to the expected frequency. The results show for the C677T, the chi-square value it was 0.07. Whereas the A1-298 polymorphism, the chi-square value it was 0.06. Both are below the theoretical value, 3.84. This indicates that there is no statistical significant deviation in the population, but they are genetic equilibrium on this polymorphism. As shown in the table, combination of genotype for the C677T and A1-298 polymorphism, the most high frequency combination were heterozygous CT genotype for the C677T and homozygous AA genotype for the A1-298C, and both heterozygous forms, CT genotype for the CT and CAC genotype for the A1-298C polymorphism. High risk combinations such as TT, CC, TT-CCM, TT-AC, and TT-CC were not identified in a group of the Albanian population. These results show that the majority of individuals carry at least one copy of the mutant allele is one or both variants. After analyzing, were compared with the mutant T allele for the C677T and T allele for the A1-298C in the Albanian population with other countries. In the Albanian population, the T allele, it was 44%, higher than more neighboring to European countries, while the T allele for the A1-298C, it was 27%, similarity to France, Germany, Italy, and Spain. The Hardy-Weinberg equilibrium analysis show that the both variants are in genetic equilibrium, not only Albania, but also like countries Macedonia, Italy, France, and Brazil. In conclusion, the finding that 56% of alleles were of the normal C variant suggests that a considerable proportion of the study population may return or near normal MTHFR and symptomatic activity. However, the remaining proportion are represented by the T alleles. Clinical significance is that both heterozygous and homozygous Curie may exhibit alternate folate metabolism influenced by nutritional and environmental factors. Thank you for your attention.
So now the second session is concluded as well. And if there is any question for all the presentations, the floor is open. And after that, we have a short break of 15 minutes, okay?
Measuring all the mutations of MTF of the gene, it's not recommended in the pregnancy. And it's nothing to do or should not be interpreted as the only cause of cardiovascular disease or every disease you mentioned. It will be in the group with others, and it's not considered thrombophilia. So this is what I have to say. Thank you.
Right, so yeah, those are really good talks. I might try to talk to some of you during the break that are here, but I was just gonna have a question for the online people. So Rafaela, if you're there.
Here I am.
Great, yeah, I was just wondering, so these variants that you identified, are these, and I might have missed this, but are these variants of interest, or do you actually know that they're causal, or suspect that they're causal?
Sorry?
These variants that you identified in the paper, do you know that they're causal, that they're actually causing the retinal disease, or are they just variants of interest?
Okay, thanks. The variant are causative. Okay.
So how do you know that they're causal?
The phenotype.
Yeah, sorry, how do you know that the variants are causal as opposed to just they happen to be shared?
I don't understand the question, sorry. Allora, I have identified the specific variant located in PRPH2 gene, and this variant are causative of specific phenotype, but this different variant may cause different type of inherited retinal dystrophy. Okay?
Okay, but you know that they cause it though?
Chiedo scusa, intervengo, faccio una rapida traduzione. Il professor Ellent voleva sapere in che modo lei conosce che queste varianti sono causative, se dalla letteratura precedente o altro.
Oh, sorry. Okay, thanks. Okay. So, allora, I identified 10 variants, okay? And 7 are published in other scientific reports, okay? Only 3 are new, okay? And these 3 variants are causative because they are present only in the subject with the ocular phenotype. So we have the possibility in some case to analyze all different subject with the ocular disorder or subject without a specific ocular disorder. Okay? But I don't have the possibility to perform a functional study.
Grazie.
Okay, okay, it's clear?
Yes, it is. Thank you.
Okay.
$15 minutes short break.
So now we are ready for the third session. I have the pleasure to invite Professor Irena Seferin. She will be holding a speech about the genotyping of erythrocyte, platelet, and leukocyte antigens, clinical applications, and first cases from NBTC.
Okay, good afternoon, everybody. It has escaped to my attention, in fact, the fact that we had to prepare our presentation in English. So my presentation is in Albanian, my slides, but I will simultaneously translate the presentation. My speech is about the genotyping of blood groups, erythrocytes, leukocytes, and thrombocytes, and I will present the first cases from National Blood Transfusion Center of Tirana. Also in the previous symposium, I presented this as a challenge of the future, but this future is now present for National Blood Transfusion Center. So since March, we are genotyping the erythrocytes for the patients in need. Okay, before going to the cases that we have genotyped, I am presenting here the two methods with which we in transfusion test blood groups. There are two, the traditional method, that is hemagglutination. We use antibodies to determine the antigens in the erythrocytes by producing reaction antigen and antibody. And this method is traditionally used, and the use of this method since many years, in fact, has demonstrated some limits in the usage of these methods. And these limits are associated to the expression of the antigens in the erythrocytes and are also associated to the status of the erythrocytes that is impacted from different diseases. And in those cases, this method is not non-conclusive. Whereas the other method that we have added to the testing of blood transfusion center this year is genotyping. And genotyping uses the DNA from leukocytes, because as we know, leukocytes are nucleated cells of blood. And so we use leukocytes to determine the genotype for erythrocytes, leukocytes, and also thrombocytes. This is the new method that we are using actually at National Blood Transfusion Center of Tirana. As you see, is from InnoTrain Diagnostics. And below, you can see the phases of testing. So from the blood sample, we go to the DNA isolation. It's 40 minutes and is completely automatic. Then the operator will do the PCR setup. It's 5 or 10 minutes. And then for 1 hour, we have the amplification and the results. So in 2 hours, we can conclude the genotype, the extended genotype of erythrocytes based in this method. And we have begun to use this method for the extended genotype of erythrocytes, and which are the patient populations that need this approach in order to determine their blood group are patients that have multiple transfusions, because in patients with multiple transfusions, when you test erythrocytes with the traditional method, you have two types of erythrocytes there, the erythrocytes of the patients and the transfused erythrocytes, and so you cannot conclude in this way. And then our patients with hemolytic autoimmune anemia. These patients have problems with their erythrocytes, which are coated with antibodies, and you cannot test. and you need genotyping. And then you need also genotyping in order to determine, in order to clarify the status of the antigen D, which is polymorphic and gives non-conclusive results with serology, with the traditional method. And of course, you can use genotyping in order to manage appropriately the conflict between mother and fetus in the hemolytic disease of the fetus and the newborn. And now here I have the cases that we have done since March of this year. We have treated 2 cases, diagnosed their extended genotype by the new method. So in the autoimmune hemolytic anemia, as you can see, the erythrocytes are coated with an antibody that the patient produces. This antibody coats the erythrocytes of the patients, and those erythrocytes are destroyed prematurely. And because of this reason, the patient has anemia. And when the patient has anemia, he needs also blood transfusion. And in order to give him transfusion, first of all, you have to determine his blood group. And in these cases, patients need to have an identical transfusion. So you have to go with an identical blood transfusion. And in this case, with these erythrocytes, with the traditional method that tests erythrocytes, in order to find the antigen in the erythrocytes, it's very difficult because everything becomes positive. You have this pan-agglutination, and you cannot decide about the phenotype of this patient. And this is our first patient that is 76 years old. And the diagnosis is lymphoid, chronic leukosis, and the complication was hemolytic autoimmune anemia. And here you have the serology. And as you can see in those 2 cases, you cannot determine there the phenotype because the agglutination, it's not conclusive. And you cannot determine the phenotype of this patient. So what are you going to transfuse to this patient? So the transfusion becomes difficult. But with the genotyping, as you can see, those circled there are the antigen E and the antigen Cal. And with the genotyping here, we managed to determine the phenotype. As you can see, the E, the patient lacks the E, and the patient lacks also Cal. So with this, we determined the extended genotype of the patient, and so we can guarantee an identical transfusion to this patient. Also, similar is the other case. You can see here the serology. All the results that are circled there are inconclusive results. So with serology, you cannot transfuse this patient. And with genotyping, as you can see here, we have clarified the situation of the patients related to the all antigens in rhesus blood group, in KID blood group, in Duffy blood group, and it's also in MNS blood group. So this is what we have, the cases that we have treated since March, but what are we going to do in the near future and also beyond with the extended genotype? In the near future, so this year, we will begin to test all our donors that test negative in serology. We will test with genotyping. Why? Because as you can see here, we have As you can see here, this is a rhesus positive, D positive erythrocyte, and this one is negative erythrocyte. Here, this in the middle is the del phenotype, which is where the antigen D has a very low expression. And when you test it with serology, this is negative. But when you transfuse this blood to a patient that is negative, this antigen here, even if low expressed, can immunize the patients, and the patient can develop antibodies anti-D. So we will pass all our donors that are rhesus negative in serology. We will do the genotyping so that we discover these cases that, even if rare, they can cause immunization in the patients. And this is one of the group of cases that we will treat this year. And then we will treat also all cases that are weak serology, women, and also patients. Here we have what we actually do with pregnant women and patients that have a serologically weak D. Actually, what we do, we treat them as negative. Even if they are serologically weak, we treat them as negative. So the pregnant women, we give the anti-D IgG as prophylaxis. Whereas the patient that is serologically D negative, we transfuse with blood negative, rhesus negative, and in this case, we have a negative impact in our stock of blood. Because if we genotype these patients and these pregnant women, in 82% of cases, this will be positive and will be treated as positive, and also with transfusions as positive. And in the remaining 18%, they will remain negative and will be transfused and treated as negative. So this will positively impact our stock of rhesus negative blood. And then in the future, we can also go on with the genotyping of leukocytes. We already can do it in our National Blood Transfusion Center, but it needs a specific budget. And with extended genotype for leukocytes, of course, we will support the transplant of hematopoietic peripheric stem cells. We already have had in Albania an autologous case of hematopoietic peripheric stem cell transplantation, but also when we go to the homolog hematopoietic stem cell transplantation, we will need the compatibility in the leukocytes, in the genotype of leukocytes. And we actually can determine all the antigens of leukocytes with this method that we have in use actually in transfusion center. And of course, we can determine also all the antigens of platelets in order to treat the thrombocytopenic post-transfusion purpura with identical transfusion. So future application of genotyping in Albania will help us in creating a national registry for HLA, for HPA, and also for erythrocytes, and will help in the prevention of the hemolytic disease of the newborn and correct evaluation of the risk, in a better treatment for multi-transfused patients. It will reduce the immunization, the alloimmune immunization, and the complications of transfusions. and will help us in a personalized strategy of transfusion, and of course will support the scientific developments in the National Blood Transfusion Center. So thank you for your attention.
Okay. Thank you, Professor Safari, for the very interesting presentation. Now I'm going to present myself. I'm Emilio Gene. I'm a biologist, embryologist. We will go on talking about pregnant women, hopefully pregnant, and babies. And nevertheless, the beautiful chants from the next door mosque. I'll try to talk about my presentation today that will be about the PGTA analysis. That is the most interesting genetic part of our job. That means that it's the pre-implantation genetic testing for aneuploidies that we do in the embryos previous to the transfer. And my question in this presentation was whether we are improving the overall outcomes of our job or just refining the selection of the embryos. So why this analysis matters, why it's important eventually to do the PGTA test at the blastocyst stage of the embryos. This is thought to improve the embryo selection by specifically choosing only the best and normal embryos. And as a consequence, it can reduce the miscarriage rate, it can reduce the time to pregnancy, and also increase at last the live birth rate. But the main question remains, does this PGTA analysis truly improve our clinical outcomes? So let's see, here is a short representation just in order to explain you how this functions. We have the embryo, we hold it from the left side with a very small pipette. This is day 5 or 6 of the development of the embryo, and we do some small holes with a laser that you are not able to see here in order to get just a small amount of cells, that it might be around 8 cells more or less. And this here is the final portion that we collect, put in a tube, and send to the genetic lab in order to analyze. I also have a short video here to show to you, just in order to understand from a practical point of view, how do we do this test. I know that this seems a little bit too much, but this is the way we do. We try, of course, not to touch the inner cell mass that is on the other side of the embryo. And after that, the embryo is frozen in the liquid nitrogen tanks, and we decide to sew it back. If the embryo is fine, it's okay. And the cells re-expand, so the embryo is still fine also after this analysis. So what PGTA actually promises? It promises to detect aneuploidies by selecting, of course, only the euploid embryos. It improves selection, but not necessarily the biology itself of the embryo. So PGTA does not actually create the better embryo because the embryo is the one that we have. It just helps us to select the best one among them. We also have some limitations, of course. First of all, we have biological limitations because we can face the mosaicism effect. That means that the trophectoderm cells that we are collecting, that are these cells around here, maybe it's not said that they are representative of the whole embryo. So we could have a risk of false positive or negative. The embryo itself has a self-correction mechanism, so some aneuploidies can still develop normally. And also we do not have access to all genetics because we have no specific information regarding single gene disorder of that embryo or epigenetic defects. For this thing, we must add another kind of analysis. We might face technical limitations as sampling errors or platform variability because the analysis that is used for testing this kind of cells is the NGS nowadays, but different labs have different thresholders, and maybe this can lead to inconsistent classification. And also, we don't have at the moment a universal reporting standard specifically for the mosaic embryos. Other limitations are the one of clinical nature. Perhaps this could have an uncertain impact on live birth rate. The main category that we think that can benefit of this technique is the group of advanced maternal age patients. There might be a chance of possible loss of viable embryos, and this leads to an overselection. So the overselection may reduce the cumulative pregnancy rate because by having some of the embryos aneuploid, we just discard them. And of course, we don't have a guarantee of the implantation after the transfer of these embryos or of healthy birth. There are some risks related to the procedure, like the embryo can be stressed. That's why it's a very delicate technique, and it requires really a very well-trained staff to do that. We have to consider the freezing and thawing procedures after that, as I mentioned before, and also some added costs at one time. Because here in Albania, the government does not cover the expenses, so the patients have to pay for the IVF cycle if done in private centers, because now there is 2 years that we also have the public center doing IVF, but they don't do this kind of test. And they have to pay extra for the drugs and for the genetic test, so it's a lot of added costs. And at last, we might also face some interpretation issues, like the binary result, euploid or aneuploid, is just sometimes an oversimplification of the biology itself of the cell, and also limited feedback from clinics that are sometimes hard to validate. So before going to see into our data, into our study, I just selected an interesting work in literature. This is a very big study done of 3 groups that put it together, their effort, their patients, their numbers, in order to give us really a strong statistic overview of the situation when PGTA test is applied to the blastocyst. So this is a multi-center analysis done from a clinic in Spain, in Abu Dhabi, and the last group is the one from Boston IVF. They went through 40,308 blastocyst biopsies, so it's really a huge number. Only 8,491 were the ones that were euploid and transferred. And this, of course, were their real data from daily work in the lab. The mean age of these patients analyzed was around 35.8, and the euploidy rate of the overall embryos analyzed was 53%. The live birth rate was stable across age group. It was around 45% under 42 years old women and around 34% over 42 years old women. So basically, they divided this huge study in 3 groups based on the 3 different clinics, and they noticed that there are different interpretation criteria among the 3 groups. The first group perhaps had standard euploid rate of the embryos analyzed and standard live birth rate. But on the other hand, the second group had more euploid embryos after the analysis, but the live birth rate was lower. So this is a strange thing. The third group had fewer euploid embryos after analysis, but the live birth rate was comparable to the first group. So that's to show how different clinics apply this technique in different ways, and this leads to different results. So the live birth rate depends highly on the selection criteria of the embryos, because it's also important to select good embryos to analyze, on the lab strategy, and not only based on the embryo biology. So same embryos, different decisions, different outcomes. As a key finding, we can say that PGTA is significantly influenced by embryo selection strategy. The live birth rate vary between centers and also between different labs. So what we have from the evidence from literature is that there are at the moment no universal standards for the mosaicism threshold. That is really a delicate argument. And for the embryo prioritization. So same technology, always using NGS, next generation sequencing. We have different outcomes among different clinics. So take home message, PGTA effectiveness is not only technology dependent, but it's strongly influenced by the laboratory practice and the clinical interpretation. So now let's go to our study. We don't have the opportunity to have that big numbers because, as we said before, we could have also more and more patients doing this test, and it could be really beneficial. But at the end of the day, it's also a matter of cost. So not all of the patients can afford to go through this. Our objective was to evaluate the real-world data coming from our center and to assess whether the PGTA analysis based embryo selection translates into improved clinical outcomes at the end of the day. So we had the chance to analyze 111 PGTA cycles for overall number of 317 embryos at the blastocyst stage. That could be day 5 or day 6. All the patients then underwent to single embryo transfer. NGS was used also in our case, and all the embryos were created after the ICSI procedure, mean maternal age 37.3. The indications for the group of patients selected was the advanced maternal age and the recurrent pregnancy loss. These are our main outcomes after the study. So we had the chance to transfer 63% of the embryos, and the euploidy rate of the analyzed embryos was 54%, aneuploidy rate as a consequence 46%. We also had patients with all aneuploid embryos, so this was important because they might have transferred aneuploid embryos, and in this case, of course, it was critical because they were not able to make a transfer at all, but at least they had an information. Clinical pregnancy rate of the transferred embryos was 59%. We also had miscarriage rate 12%, live birth rate 38% so far, but we also have some ongoing pregnancies. So overall, live birth rate and ongoing pregnancies makes 47%, which is really a good number. We wanted to compare ourselves to the rest of the world. So what's going on here and what literature is saying. So in this table, we saw that the euploidy rate of the embryos analyzed was almost the same. We got a total euploidy rate of 54%, sorry. And in the study that we saw before, they had an aneuploidy rate of 53%. Aneuploidy rate as a consequence was similar, and all aneuploid embryos from one patient, in our case, was 27%, and this was a criteria affected by the classification. We had a transfer rate about 63%, and also this depends on the euploid labeling embryos, but it was like an efficient selection. Clinical pregnancy rate, it was 59 in our case, and it was similar also in the other study across strategies when the embryo is euploid. Miscarriage rate, also it's predictable in this group of patients. And also the live birth rate, it was really very similar in both cases. Observation. So our results are in line with an intermediate PGTA approach. Then the data shows a balanced euploidy rate with strong clinical outcomes supporting an accurate selection rather than overclassification. So if we use a permissive criteria in selecting the embryos, we increase, of course, the transfer opportunities, but this may affect the success rate. If we use, on the other hand, the strict criteria in selecting the embryos, this can improve the outcome, but it limits the overall number of embryos that we have available for the patient. And the embryo quality, as I mentioned before, and biopsy day has a significant impact on the chance of live birth rate, because of course we know that day 5 embryos are better compared to the one of day 6 or day 7. It's really very rare to use them. So this analysis, it really makes sense and makes the difference in the advanced maternal age group, in the patients that had recurrent miscarriage and maybe also repeated IVF failure cycles. We have to pay attention when we offer this option to the young patients, to the good prognosis group, or to the ones that are facing this problematic for the very first time, and they are just doing the first IVF cycle. Because really in these cases, data shown that it doesn't make a difference. Take-home message, so PGTA, it's not universally beneficial. We still have to deal with the mosaicism challenge. So we need, sometimes maybe we can need a better selection in this case with the AI that we have, we might have available. And nowadays, it's always more and more common, maybe with specific programs that can do embryo selection or also with multi-omics. PGTA may improve selection, but not necessarily outcomes, as we said, in all patients. It's very patient-specific, and the patient selection is exactly the key. And the real-world data are not always aligned with the expectation. So as a final thought, I can say that PGTA is just a selection tool, but not really a guarantee. More information does not always mean better outcomes, so we really have to pay attention, and the clinical judgment remains really essential. This is our group, thanks to all these beautiful doctors and biologists. We have the chance to change the life of the people every day and to make them happy, bringing kids and creating beautiful families. So just a big thank you for your attention. And after this questions at the end, I would like to introduce the next speaker, that is Adela Perola, talking about clonal hematopoiesis, the genetic bridge between aging, inflammation, and systemic disease.
Thank you very much for being here, and thank you very much, Professor, for inviting me here in this international symposium. Thank you very much. I really appreciate it. The idea of studying clonal hematopoiesis and if there is a genetic bridge between aging, inflammation, and systemic disease, in fact, it was started from a study of European Hematological Association scientific working group that is studying also the clonal hematopoiesis, like the bridge, the really bridge of the age, inflammation, and systemic disease. They are all linked through clonal hematopoiesis. So starting from that, me and a group of my colleagues and the residents, we have done like a narrative review of the literature until now published in order to follow up with other real results and a real good study on that. I have nothing to disclosure, and what is studied or what is seen until now is that aging is the process linked with the starting of somatic mutations in hematopoietic stem cells, and after that following with a clonal expansion of those hematopoietic, malignant hematopoietic stem cells that are more fit comparing to the normal hematopoietic stem cells, and giving so the the situation of a pro-inflammatory state of the milieu where those cells are on the niche of the hematopoietic stem cells, and starting all the systemic disease, not only the hematological malignancies, but all the other hematological diseases. And what is accused, it's just clonal hematopoiesis that starts this chronic inflammation, and they accuse the accumulation of somatic mutations from those genes that control epigenetics. And what we have myelodysplastic syndromes, we have acute leukemias, we have multiple myeloma that is based on B-cell lymphoma activated and transformed in monoclonal plasma cells. And but also we have non-malignant systemic diseases like cardiovascular disease, autoimmune disease, and also osteoporosis and cognitive decrease and decline. First of all, I have to say that the images are done with artificial intelligence. So just I have put it all the results we have together and put it in the artificial intelligence. It's an open artificial intelligence. And what we have questioned in the consensus artificial intelligence, the question is, it's for real that clonal hematopoiesis is really the bridge between aging and other diseases. And 79% have been accused that, okay, yes, the studies have shown that, okay, this clonal hematopoiesis is the cause of not only hematological malignancies, but also non-hematological malignancies. So we have used STITA-I, is also an artificial intelligence tool that have searched more than 170,000 and more studies that are published in all the databases, but focused in PubMed, focused also in other, for example, Web of Science, and we have chosen from them only the 50 more cited and the 50 more important papers published. And what have we seen? We have seen that there are not so much publications about the clonal hematopoiesis like a bridge or like the cause of inducing aging, inducing also inflammatory process and non-hematological and hematological diseases. And I don't know if it's good look, those that are the larger are the more cited, like the publications more cited. So what are the results? The first result, it was the prevalence and about the prevalence and genetic drivers of clonal hematopoiesis. And all the studies demonstrate that before the age 40, we don't have a clonal hematopoiesis. It's extremely rare. But after that, you can see a sharp rise in the level of clonal hematopoiesis with the maximum about 20% present in the population that are more than 80 years old. In the other hand, it will be that are those mutations that are epigenetic regulators, DNMT3A, TET2, and ASXL1, that are the more accused, and there are about 90% of the cause of all the clonal hematopoiesis and deriving after that all the diseases. But with 50% founded of this regulator, that is DNMT3A, 20 to 25% has been found with TET2 mutations, and also with a little until 5% of ASXL1. Speaking about JAK2 signaling and TP53 mutations, that was about 5%. So the clonal hematopoiesis, it's really important biologically because it's an age-related process linking somatic mutation acquisition with the stem cell selection, and so giving rise to hematological and non-hematological diseases. The second result that we have found it from those 50 articles, it was that those are based on, sorry, about that. Based on the publication of Belizer 2023, Kaleido in 2022, and also of Wellem and 2025, we have seen that the most important thing we have to show that those TET2 and DMT3A mutation are the most important that give rise to the of the clonal hematopoietic stem cell in the bone marrow, and those clonal hematopoietic stem cells, or this clonal hematopoiesis, give rise of mutant monocytes and macrophages that produce elevated interleukin-1 beta and interleukin-6 that are really important on maintaining chronic inflammation. That it's inflammaging. The term it's used, inflammaging. More we age, the more we have those products. More we have, the more we age, the more we have mutant monocytes and macrophages that give rise to this elevated production of chemokine expression and also inflammasome activation. And this secondary give rise to atherosclerosis and also to immune dysfunction. And we have, for example, rheumatoid arthritis, COPD, or psoriasis. And on the other hand, this chronic inflammation comes back to maintain and augment also this normal hematopoiesis. The third finding we have from those studies we have studied, it is that mostly we have found a systemic disease association with the mutation found, and we have the TET2 correlation strongly linked with cardiovascular disease via inflammatory mechanism. So if we find those, we have to see also the strong correlation with cardiovascular disease. We have also found that it's a strongly correlation between the mutation of DNMT3A with heart failure and osteoporosis. And we can find people with osteoporosis having heart failure or vice versa. And also the SLX-L1 mutation has a strong pro-inflammatory signature, but in the face of only 5% of findings, there are more to be studied on that. But as you see here, the clonal hematopoiesis give rise to all the cause mortality, to autoimmune disease, to osteoporosis, to gut or podagra, to solid tumors, and others, cognitive impairment and dementia, and chronic renal disease. So while we age, we create this clonal hematopoiesis, and it's all that explained. But we have this bidirectional feedback. So in one hand, we have this chronic inflammation that gives rise to clonal hematopoiesis expansion, and this clonal hematopoiesis expansion, due to the fact that we have these mutant macrophages and monocytes, increase the chronic inflammation. So we have a loop. And while we have this loop, we have the consequences. So we have the diseases. What are the studies that claim what we have found it? And we have, I have listed here the evidence strength of each publication we have studied. And in this case, the strong publication or the strongest evidence, it was from, it was Jaiswal and Libby, Asada Kitamura, and Jaiswal Ebert in the studies of 2019. And it was also from Belizer in 2023, Jaiswal and Libby and Policio in 2023, and also from Liu in 2025 and Kayado in 2022. And what is strong evidence? It is that clonal hematopoiesis increased with ages due to somatic mutation in hematopoietic stem cell, and those mutant clones drive chronic inflammation via altered immune cell function. On the other hand, another strong evidence is that the clonal hematopoiesis is an independent risk for cardiovascular disease and all-cause mortality. And on the other hand, chronic inflammation promotes the expansion of those mutant hematopoiesis, of this mutant hematopoietic stem cell, and this, it's a feedback loop. And we have a moderate, also moderate evidence that will be that mutation-specific effect can modulate the risk and severity of different diseases. And on the other hand, the clonal hematopoiesis in non-malignant diseases still remain incomplete, and we have to focus on that. So the key interpretive note of what we have found are that the relationship is bidirectional, and the clonal hematopoiesis can promote the inflammation-driving disease. On the other hand, mutation specifics are TET2 that may drive stronger inflammatory responses than DNMT3 or ASLX1 in some context. On the other hand, we have a problem that not all the individuals that have high level of those mutations can develop disease, and this is okay, telling the truth. Why someone can have higher burden of those mutations and never develop a disease, and on the other hand, we have some other people that have mutations but develop the most severe form of the disease. Those are the discussions. So the evidence support clonal hematopoiesis as a genetic bridge, and also this clonal hematopoiesis increase the risk for cardiovascular disease and mortality. And the other hand, that we have specific drive mutations that induce pro-inflammatory phenotype. So not all, the problem that I have linked here with the yellow is that not all individuals with clonal hematopoiesis develop this overt disease. And this is the gate that we have to study. Those are the conclusions, but for me, the most important thing to be mentioned is that it's a bidirectional relationship that can create a self-perpetuating cycle and drive mobility among all across organ systems. Those are the research gates, so the field that we have to study in order to really understand that clonal hematopoiesis induce diseases, and what are the questions for the future to be studied in clinical trials are that why some individuals with large mutant clones remain free from disease, and second, what is the precise molecular mechanism that can underlie mutation-specific effect in those patients, and can those, this is really important for us as clinicians, do we have target anti-inflammatory therapies in order to reduce morbidity and mortality? Because everything we are studying, everything we are doing is that in order to cure disease, to prevent them, or to have a life without disease, what it could be the best. And aging only like age, but without disease. Those are the references, so I'm passing through. So thank you very much, and I'm open to questions.
Okay, so thank you, Professor Parola, for the very interesting presentation. Maybe we would have some questions at the end. Now let's move to the next speaker, Lenida Sure. She will be talking about the phenotypic and genotypic discordance in the diagnosis of drug-resistant tuberculosis. And she's from the Regional Hospital Center, Shefqet Ndroqi.
Hello to everyone. Thank you, Professor Lia, for the invitation. I will discuss now the evaluation of the genotype and PPR plus, or line-for-the-say method, and phenotypic, genotypic discordances between these two tools in drug-resistant tuberculosis. I will present our work in the National TB Reference Lab in our country. The diagnosis of drug-resistant tuberculosis is very important
The first point is the successful treatment of the patient, the lack of the patient, the first point. And after that, if we have an early identification of resistant cases, we can control better the spread of infection to interrupt the chain of transmission into in population. And the most important thing is to preserve the effectiveness of anti-tuberculosis drugs. Why I'm saying this? Because we have a few drugs that work against tuberculosis. If for pneumonia, let's say we have some classes that can affect or can we can use to pneumonia. For tuberculosis, we don't have respiratory. So an appropriate diagnosis and fast diagnosis is crucial for the life of patient. And the other important thing is that tuberculosis has an extended time to grow up. If we have a sample today, we have to wait until 2 months to say if this patient is negative in the most extreme time. If positive, maybe for 3, the best time, to 6 until 8 weeks. So the time is a crucial factor for TB. This is the early algorithm that is used in the most of TB European laboratories to diagnose and follow up the TB and MDR TB cases. We use in our lab too, but I have simplified in this chart to explain the steps that we follow up in our lab. The first step, so the first step is the sample collection, like in every bacteriological, microbiological analysis, and it has a pre-analytic condition, like have to be in lab into 2 hours and other like this. I don't want to waste time. After this, the first step is Zellnitz staining. When we see in microscope if we have the presence or not to mycobacteria. Parallel in this, we do the gene expert test, that is in PCR technique, that we detect if we have the presence of mycobacteria into the sample. And this test gives us the information about rifampicin. That is the most important drug against TB. And after that, if we have FEB stain positive, we perform blind probe assay first line and sometimes second line. After that, all the samples, positive, negatives, go to culture in solid media and liquid media. So this is the part that I said is most extended part. 8 weeks. To say sample is negative for TB, we have to have the culture negative. This is the culture is the gold standard for all the samples that are into our lab. And after we have a live culture, we isolate the TB in culture, we have to perform DST, drug susceptibility test, in 2 forms, phenotypically and genotypically. So what is the role of genotype and DDR plus test in the laboratory diagnosis of tuberculosis? At first, the most that we win for this test is as I said, is the time. So if we detect the presence of Hemoglobin tuberculosis directly from clinical samples. If we have to wait some weeks, in 1 to 2 days, we have a result and an information about rifampicin. And this test identifies the main genetic mutation responsible for resistance, like rifampicin, that is the main antibiotic drug that is used against TB, and isoniazid, that are 2 responsible genes for this resistance, CAG and INHA genes, and provides information
on resistance to isoniazid. Because, as I said, the first information we give for rifampicin we give from the gene expert analyze. Steps of LPA procedures are, as I said, as every genetic procedure is a sample collection. After that, it's a DNA extraction, PCR amplification, and after that, line probe assay hybridization and detection of mutation. And in the end, we give a report to clinicians to decide what antibiotic is appropriate to treat the patient. This is the phone, how like the strips is shown. There are like letter strips, let's say, paper strips. And if we have all wild type, all bands wild type shown, and all mutation is, we don't have mutations, we are talking to a sensible, or we have not resistances. If some of mutation is showing and some wild type is missing, now we can talk to mutation and to resistance from these drugs. And these photos show the application of the procedure in our lab from all the stages steps that I mentioned. Like every tool, this tool have advantages and disadvantages. The most major advantage is short time compared to culture. 1 to 2 days, we have a result that is very, very important to treat the patient. We don't have to wait for weeks. The other advantage is high sensitivity when bacterial load is high. If we have contact patient that has been in contact with TB-resistant mycobacteria, we can use this like an initial test for patients to take information for rifampicin resistance. And after that, this is a good standardization using a certified commercial kit, but has some disadvantages. That does not replace the culture. As I said, culture is the gold point in a diagnosis for TB because we want to have a live mycobacteria. To perform DST and other tests requires specialized laboratory staff, biosafety measures, so we have necessary for logistic other terms. And low sensitivity when bacterial load is low in direct microscopy and does not detect all mutation. Some resistance mechanisms may be out of the regions that we are testing for. And has higher cost than microscopy requires well-trained staff. I will show very shortly the causes of discordances. Unknown error mutation outside the target region, silent mutation with no phenotypic effect, heteroresistance, we have mixed population that under the antibiotic pressures gives priority, this phenomenon gives priority to mycobacteria that are resistant. And other are partial expression of resistance and technical or human errors during the procedure. This is the region of rifampicin resistant determining region, with 95% of this of mutations occur into this region. In our study, we have used 438 strains of Mycobacterium tuberculosis during the period 2021 to 2025, and the phenotypically results were compared to genotypically results. And phenotype DST, as I said, were compared with the line probe assay or MTBDR plus molecular test. And what do we have found? If we refer for rifampicin, if we refer phenotypic sensitivity, we have 17 cases against the genotypically sensitivity with 16 cases. Resistant, we have 14 cases, and genotypically resistant, we have 50 cases. This chart shows this, if we can see the gap is only one case, or difference between phenotypically test and genotypically test. It's not the same situation if we refer to isoniazid. The discordances between samples is 6, sensitive and resistant. This is shown in this chart. So the question, so if we analyze in total all the results, we have in discordances between phenotypic and genotypic testing, we have 45 fits in only one case, that is discordances. We have 7 cases for isoniazid, and 77.5% or 24 cases has no discordance. And the point is, the clinicians or doctors say, we have said a lot of things, but how to treat the patient? Which is the test that I have to use, or the drugs that I have to use? I have to consider phenotypic test or genotypic test. So this is the point that we have to discuss. Referring to, we have to analyze all the most important drugs that are rifampicin and isoniazid. In case of rifampicin, referring to WNGO and supra reference, also reference to upper the others. If we see a mutation into the for RPOB gene, immediately it will consider like resistant, and the patient will get like MDR patient. So always mutation in RPOB gene are consistently associated with resistance. It's not the same with isoniazid. A combination of both approaches is used. But if we catch resistance in genotypically test, we have to compare with phenotypically test. So are two different kinds of the story. For other drugs, for the first line, that is ethambutol and pyrazinamide, the phenotypically takes priority. And for the second line, that fortunately we have a few resistances in our country, maybe 3 cases per year with TB resistant, phenotypically and genotypically results are interpreted in combination. But phenotypically results have priority. And so the summary is that phenotypic DST is considered the gold standard, except for rifampicin, where genotype testing is highly reliable. This was, as I said, not to waste time. And conclusion, genotype MTBR+, or as we know, LiProbase, is a validated method for detection of drug-resistant tuberculosis. Because the rapid detection of mutation associated with MDR-TB and significantly reduced turnaround time compared to phenotypic testing. But it does not detect mutation outside the targeted regions that we, in our experience, all the time that I am in this job, we have had only 1 case. And this case was evaluated and was sent to our supranational lab in Milan, and we found that this mutation were outside the region that we study in our routine workday. And in case of that, of course, additional methods such as next-generation sequencing are recommended to improve diagnostic accuracy. This was my presentation. Thank you.
Okay, thank you, Ms. Surai, for the beautiful presentation. And now we will move forward to the last presentation. So last but not least, we will be going on to talk about tuberculosis with Ms. Blerina Rapo, and she will talk about a novel genetic tool in non-tubercular myobacteria diagnosis and identification.
Hi, it's pleasure to be here. Thank you, Professor Ilia, for the invitation. This is a little switch from another kind of mycobacteria called non-tuberculous mycobacteria. These bacteria, or NTM, these bacteria don't cause tuberculosis, but they cause serious infection, especially at immunocompromised people, and today are more frequent to be grown in labs. Finding them is very important, it's just case finding. I'm going to start with what are NTM and give you a brief epidemiology because I want to make the point that this is a very important and emerging disease. Unlike TB, if you grow one colony and it's not due to contamination, the clinicians treat everybody pretty much the same, but it's much more if they have drug-susceptible disease, but it's much more difficult with NTM than it is with TB or tuberculosis. Then I'm going to focus on the LPA, the genetic method you use to identify and to detect the NTM species. These bacteria belong to the genus Mycobacterium. There are over 200 species and subspecies. You know, bacteria that causes tuberculosis and leprosy, and all the other members in this genus are called non-tuberculous mycobacteria. On the right, there is a genomic map with 200 species, which share one common characteristic. They are called acid fastness because they retain the stain during acid exposure during staining process. The incidence is rising worldwide, especially in the developed country. If you see in the red area, the most common mycobacterium is Mycobacterium avium complex, followed by Abscessus fortuitum kansasi. Depending on the rate growth, these bacteria are divided into two large groups. Slowly growing mycobacteria are bacteria that need more than 7 days to grow, and rapidly growing mycobacteria are bacteria that need less than 7 days to grow. These bacteria are present in nature, in water, soil, dust, so the source of infection is environmental and the human does not transmit it. Tap water is considered to be the largest reservoir for NTM. And these bacteria have a lipid-rich membrane, which allow them to adhere and to colonize pipeline surfaces by forming biofilms, and so they are very resistant to disinfectant. These two slides now shows how some NTM become resistant to antibiotics. Resistance can occur either through acquired mutations or intrinsic mechanisms. Here are the three more important genes. Each one can cause resistance in a different way. For example, mutation in RRL gene prevent macrolide antibiotic from binding to the ribosome and so leading to a strong acquired resistance. Mutation at GRA gene alter the DNA geras, so interfering at the ribosomal for fluoroquinolone antibiotics activity, and mutation in the ERM41 gene cause inducible resistance. As soon as the sample comes in our lab, we perform microscopy and culture both in liquid and solid media. If the sample from culture is positive, we perform another quick test called MPT64 antigen test. If from this test the sample is positive, we refer as MTB for tuberculosis, because MPT64 is a protein that is produced only by members of MTBC and is absent in non-tubercular mycobacteria. So if it's negative, we continue with NTM and with genotype mycobacteria CM assay with line probe assay. What is LPA or line probe assay? Is a molecular method to detect NTM and to detect resistant genes. LPA is based on the DNA amplification between the amplified mycobacterial DNA and specific probes fixed on the nitrocellulose membrane. Here. These are the reagent preparation. Here I want to highlight only two safety precautions for PCR. We have rooms separated for DNA extraction for PCR and for reverse hybridization. These are the steps for line probe assay. After DNA extraction, the DNA is amplified by PCR. The amplified DNA is labeled with, sorry. I want the red point. The amplified DNA is labeled with biotin, and then the labeled biotin single-stranded DNA is bind to complementary DNA probes here on the nitrocellulose membrane. Then the streptavidin is strongly related to biotin. Streptavidin also is linked with alkaline phosphatase, which is an enzyme that gives a color reaction. And at the end, when chromogen substrate is added, we have colored band at the end of the procedure. Here is the steps of DNA extraction. I want to only highlight the kit we use, generalized version 1 from Berger. These are the PCR amplification and master mix and the steps. This is the reverse hybridization. We used 2 incubator, and this is our strip at the end of the procedure of LPA. A valid test, in valid test, it's very important to see the 3 control bands to validate the results. On the left of the strip here, there are 3 control bands. The first one, conjugate control validates the process of hybridization. The other one, the second one, validates the process of extraction and amplification. And the genus control shows whether members of genus Mycobacterium are present in our sample. This is the interpretation chart that we use to do the identification of non-tubercular Mycobacterium. The result in a 2-year study, we had 28,000 samples from them. We had 272 culture positive. From them, we had only 16, From them, we had 16 Mycobacteria non-tuberculous divided in 4 rapidly growing and 12 slowly growing. After doing identification tests, we had this species, 1 Xenopi, 7 Specium, 2 Gordone, 1 Malmoense, 1 Avium complex, 1 Abscessus, and 3 Fortuitum. This is the distribution of cases by years and age. We have the most cases in the older age and most cases in Tirana and in males. Here I have shown a microscopic view of Mycobacterium abscessus, which cause aggressive infection in lungs, skin, and soft tissue. The main message, early diagnosis of NTM is the key to preventing lung damage and the misdiagnosis as tuberculosis and to improve the patient outcome. As for infection prevention, the use of water filters should be very used to mitigate the risk. And the decision to start antibiotic treatment should be based on 3 factors: patient, the type of mycobacterium, and the purpose of treatment. Of great importance is the creation of protocols and awareness and support for patients with NTM. And this method, genetic method, LPA, should be used in all TB reference lab until sequences will be a routine one. Thank you for attention. This is our staff.
So I would like to thank all the speakers for all the beautiful, interesting presentations, and also to Professor Elia for organizing this beautiful symposium every year. And now the floor is open to questions for all the speakers. And maybe I can start with the first question I wanted to ask to Professor Perola. So in order maybe to put together our presentations and our work, I was asking myself, if do you foresee maybe the IVF treatment, reproductive medicine in the next future being impacted by the bigger knowledge in the next years about the somatic clonal mutations eventually?
Okay, yes, I think that it's already in your work, because most of the people who are going to your clinic are more than 40 years old. Telling the truth, you are facing that just now.
Yes.
With the girls
Just wanted your opinion.
In my opinion, yes, more than 40. It's not so as 18.7 to 20% of the increase of clonal mutations going with age, but if the people that are going, the girl or the woman that are coming to your clinic or your clinics to get the IVF, you have to check for all the other diseases they can have. And also, if I could remember, because I'm not so good in my memory, used to be that the girls that get pregnant less than 35 years old have the lower risks to have mutations in the pregnancy, to have Down disease or other chromosomal diseases. So we are trying to get pregnant women that are more than 40, 45. It arrived to me for consultation, a woman that was 55, and she wanted to get pregnant. And the idea was that, do I have the blood in normal ranges in order to get the IVF? So this is a question for you.
Yes, here in Albania, actually, we have the law that we can go further after 50 years old for the women to have IVF treatment. So we are really strict to this law. Maybe not all the clinics, but we do so. And this is very important. We had a patient of 48 years old, and she wanted to have IVF treatment with her own eggs, and it was very challenging. But she had embryos, and of course we suggested her to do the PGT-A as a minimum, but she didn't want to do because she was optimistic and she wanted to try that way, an embryo transfer without the analysis. So these are the daily problems of the patient. Okay, so if there are other questions.
Also a question for Professor Perola. Yeah. Thank you for this very interesting review. My question is, does it come out in any of these studies to what extent a healthy and correct lifestyle prevents clonal hematopoiesis?
It was all mentioned in every studies. It's mentioned because epigenetics, it's you are what you eat, you are where you live and what you do. It's nothing to do with the mutations. It's epigenetics. It's an on-off gene that you have there without mutation, but just go through methylation or every other process. So it's really important. Yes, yes. Every publication, but the lifestyle is really important in epigenetics. But the colleagues that are geneticists can tell better than me on that.
I had a question for the moderator. So, I was just wondering, could you elaborate a bit on the mosaicism thing? So is the idea that you get 5 to 8 cells, I think you said, is the idea is that some show aneuploidy and some don't, and you don't know what decision to make, or what was the?
In this group of cells that we get, that is around 5, 8, 11, more or less, because you're not able really to count the exact number of cells at that point because they're all stick together. So the discordance or the mosaicism is between the trophectoderm cells and the one of the inner cell mass. So sometimes it's not said that the one of the trophectoderm are really 100% representative of the future fetus of the inner cell mass, because the trophectoderm cells are the ones that are going to develop the placenta. So maybe in that group of cells, we find most of them aneuploid, but the fetus itself is euploid. So we might have a bad judgment at the end of that embryo that could be good, but we classify as aneuploid and we discard it. So this was the mosaicism problem.
So the idea is you're not getting the right cells, or is it that you're getting a mixture and you don't
Actually,
know?
we cannot get the inner cell mass cells because otherwise we are going to take a part of the future baby and that embryo will not be viable anymore. But this kind of test is attendable, is correct. The answer around 95%. So it's mostly credible to be used at a clinical base, but there is still that small 2, 3, 5% that it might happen that it's a mosaicism problem in the final result of the test. So that's
Okay.
it.
Thank you.
Okay, if there are no more questions, we can come to the end of this long, tiring day. And in this case, I would like, first of all, to thank again our colleagues coming from abroad. Obviously, all the others that presented really good works. And really, I'm very happy for, especially for young researchers, that in a very short time, they have had good results. And especially because some of them have been my ex-students. So I thank you again, all the participants, to be so patient to attend this tiring day. And I wish to meet you again in the next activity of the same theme of research. So thank you again. And at the end, but not the least, I would like to thank the other part, so the administration that supports us, but we don't see. However, we got this strong support from the administration, and this is really a part of us. Thank you again, and see you in the next activities. Thank you.